Biologists have long debated whether animals arose once or several times from protist ancestors, but enough molecular and morphological evidence has now been assembled to indicate that, with the possible exception of sponges porifera, the kingdom Animalia is a monophyletic group. That is, all animals are descendants of a single ancestral lineage. This conclusion is supported by the fact that all animals share several derived traits. Similarities in their small subunit ribosomal RNAs. Similarities in their Hox genes. Special types of cell-cell junctions, tight junctions, desmosomes, and gap junctions. A common set of extracellular matrix molecules, including collagen. Animals evolved from colonial flagellated protists as a result of division of labor among their aggregated cells. Within the ancestral colonies of cells, perhaps analogous to those still existing in the chlorophyte ball box or some colonial choanoflagellates, some cells became specialized for movement, others became specialized for nutrition, and still others differentiated into gametes. Once this specialization by function had begun, working groups of cells continued to differentiate while improving their coordination with other groups of cells. Such coordinated groups of cells evolved into the larger and more complex organisms that we now call animals. Animals are multicellular heterotrophs. What traits characterize the animals? In contrast to the bacteria, archaea, and most protists, all animals are multicellular. Unlike plants, animals must take and preform organic molecules because they cannot synthesize them from inorganic chemicals. They acquire these organic molecules by ingesting other organisms or their products, either living or dead, and digesting them inside their bodies. Thus animals are heterotrophs. Most animals have circulatory systems that take up O2, get rid of CO2, and carry nutrients from their guts to other body tissues. To acquire food, animals must expend energy either to move through the environment and position themselves where food will pass close to them, or to move the environment and the food it contains to them. The foods animals ingest include most other members of the animal kingdom as well as members of all other kingdoms. Much of the diversity of animal sizes and shapes evolved as animals acquired the ability to capture and eat many different kinds of food and to avoid becoming food for other animals. The need to locate food has favored the evolution of sensory structures to provide animals with detailed information about their environment and nervous systems to receive and coordinate the information. The accounts in this chapter and the following two chapters serve as an orientation to the major groups of animals, their similarities and differences, and the evolutionary pathways that resulted in the current richness of animal lineages and species. But how do biologists infer evolutionary relationships among animals? Several traits show evolutionary relationships among animals. Biologists use a variety of traits in their efforts to infer animal phylogenies. Clues to these relationships are found in the fossil record, in patterns of embryonic development, in the comparative morphology and physiology of living and fossil animals, and in the structure of animal molecules, especially small subunit rRNAs and mitochondrial genes. Using this wide variety of comparative data, zoologists concluded that sponges, cnidarians, and ctenophores separated from the remaining animal lineages early in evolutionary history. Biologists have divided the remaining animals into two major lineages, the protozoans and the deuterostomes. Figure 1 shows the postulated order of divergence of the major animal groups that we will use in these three chapters. Several differences in patterns of embryonic development provide clues to animal phylogeny. During the development of an animal from a single cell zygote to a multicellular adult, distinct layers of cells form. The embryos of diploblastic animals have only two of these cell layers, an outer ectoderm and an inner endoderm. The embryos of triploblastic animals have, in addition to ectoderm and endoderm, a third layer, the mesoderm, which lies between the ectoderm and the endoderm. The existence of three cell layers distinguishes the protozoans and deuterostomes from those groups of simple animals that diverged from them earlier. During early development in many animals, a cavity forms in a spherical embryo. The opening of this cavity is called the blastopore. Among the protozoans, from the Greek, mouth first, the mouth arises from the blastopore, the anus forms later. Among the deuterostomes, mouth second, the blastopore becomes the anus, the mouth forms later. In the common ancestor of the protozoans and deuterostomes, the pattern of early cleavage of the fertilized egg was radial. This cleavage pattern persisted during the evolution of the deuterostomes and in many protostomate lineages, but spiral cleavage evolved in one major protostomate lineage, as we will see. Body plans, basic structural designs. The general structure of an animal, its organ systems, and the integrated functioning of its parts are known as its body plan. A fundamental aspect of an animal's body plan is its overall shape, described in part by its symmetry. A symmetrical animal can be divided along at least one plane into similar halves. Animals that have no plane of symmetry are said to be asymmetrical. Many sponges are asymmetrical, but most animals have some kind of symmetry. The simplest form of symmetry is spherical symmetry, in which body parts radiate out from a central point. An infinite number of planes passing through the central point can divide a spherically symmetrical organism into similar halves. Spherical symmetry is widespread among the protists, but most animals possess other forms of symmetry. An organism with radial symmetry has one main axis around which its body parts are arranged. A perfectly radially symmetrical animal can be divided into similar halves by any plane that contains the main axis. Some simple sponges and a few other animals, such as sea anemones, figure 2a, have radial symmetry. Most radially symmetrical animals are slightly modified so that fewer planes can divide them into identical halves. Two animal phyla, Cnidaria and Ctenophora, are composed primarily of radially symmetrical animals. These animals move slowly or not at all. Bilateral symmetry is a common characteristic of animals that move rapidly through their environments. A bilaterally symmetrical animal can be divided into mirror images, left and right sides, by a single plane that passes through the dorsoventral midline of its body from the front anterior to the back posterior and figure 2b. A plane at right angles to the first one divides the body into two dissimilar sides. The back side of a bilaterally symmetrical animal is its dorsal surface. The belly side is its ventral surface. Bilateral symmetry is strongly correlated with cephalization, the concentration of sensory organs and nervous tissues in the head of the anterior end of the animal. Cephalization is favored because the anterior end of a freely moving animal typically encounters new environments first. Fluid-filled spaces, called body cavities, lie between the ectoderm and endoderm of most protozoans and deuterostomes. The type of body cavity an animal has strongly influences the way it moves. Animals that lack an enclosed body cavity are called equivalents. In these animals, the space between the gut and the body wall is filled with masses of cells called mesenchyme, figure 3a. Pseudocoelonate animals have a body cavity called the pseudocele, a liquid filled space in which many of the internal organs are suspended. Their control over body shape is proved because the pseudocele has muscles only on its outside. There is no inner layer of muscle surrounding the organs, figure 3b. Coelonate animals have a coelom, a body cavity that develops within the mesoderm. It is lined with a special structure called the peritoneum and is enclosed on both the inside and the outside by muscles, figure 3c. The fluid-filled body cavities of simple animals function as hydrostatic skeletons. Because fluids are relatively incompressible, they move to another part of the cavity when the muscles surrounding them contract. If the body tissues around the cavity are flexible, fluids squeezed out of one region can cause some other region to expand. The moving fluids can thus move specific body parts. If a temporary attachment can be made to the substratum, the whole animals can move from one place to another. In animals that have both circular muscles encircling the body and longitudinal muscles running along the length of the body, the action of these antagonistic muscles on the fluid-filled body cavity gives the animal even greater control over its movement. A coelonate animal has better control over the movement of the fluids in its body cavity than does a pseudocoelonate animal, but its control is further improved if the coelom is separated into compartments or segments. Then muscles in each individual segment can change its shape independently of the other segments. Segmentation of the coelom evolved several different times among both protozoans and deuterostomes. Other forms of skeletons developed in many animal lineages, either as substitutes for or in combination with hydrostatic skeletons. Some skeletons are internal, such as vertebrate bones, others are external, such as lobster shells. Some external skeletons consist of a single element, snail shells, others have two elements, clam shells, and still others have many elements, centipedes. Sponges, loosely organized animals. The lineage leading to modern sponges separated from the lineage leading to all other animals very early during animal evolution. The difference between protist colonies and simple multicellular animals is that the animal cells are differentiated and their activities are coordinated. However, sponge cells do not form true organs. Sponges, phylum Porifera, from the Latin, por bearers, the simplest of animals, are sessile. They live attached to the substratum and do not move about. The body plan of all sponges, even large ones, which may reach more than a meter in length, is an aggregation of cells built around the water canal system. Feeding cells called choanocytes line the inside of the internal chambers. These cells, with a collar of microscopic villi and a single flagellum, bear a striking resemblance to choanoflagellates. By beating the flagella, choanocytes cause the surrounding water to flow through the animal. The water, along with any food particles it contains, enters by way of small pores and passes into the water canals, where food particles are captured by the choanocytes. Water then exits through one or more larger openings called oscula, figure 4. Between the thin epidermis and the choanocytes is another layer of cells, some of which are similar to amoebas and move about within the body. A supporting skeleton is also present, in the form of simple or branching spines, called spicules, and often an elastic, complex, network of fibers. A few species of sponges are carnivores that trap prey on hook-shaped spicules that protrude from the body surface. Sponges also have an extracellular matrix, composed of collagens, adhesive glycoproteins, and other molecules that holds the cells together. This molecular adhesion system may also be involved in cell-cell signaling. Thus, sponges are functionally more complex than a superficial look at their morphology might suggest. Nonetheless, sponges are loosely organized. Even if a sponge is completely dissociated by being strained through a filter, its cells can reassemble into a new sponge. Most of the 5,500 species of sponges are marine animals. Only about 50 species live in freshwater. Sponges come in a wide variety of sizes and shapes that are adapted to different movement patterns of water. Sponges living in intertidal or shallow subtidal environments, where they are subjected to strong wave action, hug the substratum. Many sponges that live in calm waters are simple, with a single large osculum on top of the body. Most sponges that live in slowly flowing water are flattened and oriented at right angles to the direction of current flow. They intercept water and the prey it contains as it flows past them. Sponges reproduce both sexually and asexually. In most species, a single individual produces both eggs and sperm, but individuals do not self-fertilize. Water currents carry sperm from one individual to another. Asexual reproduction is by budding and fragmentation. Cnidarians, two cell layers and blind guts. Animals in all phyla other than Porifera have distinct cell layers and symmetrical bodies. The next lineage to diverge from the main line of animal evolution after the sponges led to a phylum of animals called the cnidarians, phylum Cnidaria. These animals are diploblastic, have two cell layers, and have a blind gut with only one entrance, the mouth anus. Despite their relative structural simplicity, cnidarians have structural molecules such as collagen, actin, and myosin, and homeobox genes. Cnidarians are simple but specialized carnivores. Cnidarians appeared early in evolutionary history and radiated in the late Precambrian. About 11,000 cnidarian species, jellyfish, sea anemones, corals, and hydrozoans, live today, all but a few in the oceans. The smallest cnidarians can hardly be seen without a microscope. The largest known jellyfish is 2.5 meters in diameter. All cnidarians are carnivores. Some gain additional nutrition from photosynthetic endosymbionts. The cnidarian body plan combines a low metabolic rate with the ability to capture large prey. These traits allow cnidarians to survive in environments where encounters with prey are infrequent. All cnidarians possess tentacles covered with cnidocytes, specialized cells that contain stinging organelles called nematocysts, which can inject toxins into the prey. Figure 5. Cnidocytes allow cnidarians to capture large and complex prey, which are carried into the mouth by retracting the tentacles. Nematocysts are responsible for the stings that some jellyfish inflict on human swimmers. The cnidarian body is based on a sac plan, in which the mouth is connected to a blind sac called the gastrovascular cavity. The sac functions in digestion, circulation, and gas exchange and acts as a hydrostatic skeleton. The single opening serves as both mouth and anus. Cnidarians also have epithelial cells with muscle fibers whose contractions enable the animals to move, as well as simple nerve nets that integrate their body activities. Cnidarian life cycles have two stages. The generalized cnidarian life cycle has two distinct stages. Figure 6. Although many species lack one of these stages. The sessile polyp stage has a cylindrical stalk attached to the substratum. Tentacles surround the mouth, anus located at the end opposite from the stalk. Individual polyps may reproduce by budding, thereby forming a colony. The medusa plural, medusae, is a free-swimming stage shaped like a bell or an umbrella. It typically floats with its mouth and tentacles facing downward. Medusae of many species produce eggs and sperm and release them into the water. When an egg is fertilized, it develops into a free-swimming, ciliated larva called a planula, which eventually settles to the bottom and develops into a polyp. Although the polyp and medusa stages appear very different, they share a similar body plan. A medusa is essentially a polyp without a stalk. Most of the outward differences between polyps and medusae are due to the mesoglia, an internal mass of gel-like material that lies between the two cell layers. The mesoglia contains few cells and has a low metabolic rate. In polyps, the mesoglia is usually thin. In medusae, it is very thick, constituting the bulk of the animal. Anthozoans. All 6,000 species of sea anemones and corals that constitute the anthozoans class Anthozoa are marine animals. Evidence from morphology, rRNA, and mitochondrial genes suggests that the anthozoans, which lack the medusa stage, are sister to the other classes of cnidarians, and that the medusa stage evolved after the anthozoa diverged from those other lineages. In the anthozoans, the polyp produces eggs and sperm, and the fertilized egg develops into a planula that develops directly into another polyp. Many species can also reproduce asexually by budding or fission. Sea anemones are solitary. They are widespread in both warm and cold ocean waters. Many sea anemones are able to crawl slowly on the discs with which they attach themselves to the substratum. A few species can swim and some can burrow. Sea pens, by contrast, are sessile and colonial. Each colony consists of at least two different kinds of polyps. The primary polyp has a lower portion anchored in the bottom sediment and a branched upper portion that projects above the substratum. Along the upper portion, the primary polyp produces smaller secondary polyps by budding. Some of these secondary polyps can differentiate into feeding polyps, while others circulate water through the colony. Corals also are usually sessile and colonial. The polyps of most corals form a skeleton by secreting a matrix of organic molecules upon which they deposit calcium carbonate, which forms the eventual skeleton of the coral colony. The forms of coral skeletons are species-specific and highly diverse. The common names of coral groups, horn corals, brain corals, staghorn corals, and organ pipe corals, among others, describe their appearance. As a coral colony grows, old polyps die, but their calcareous skeletons remain. The living members form a layer on top of a growing bank of skeletal remains, eventually forming chains of islands and reefs. The Great Barrier Reef along the northeastern coast of Australia is a system of coral formations more than 2,000 kilometers long and as wide as 150 kilometers. A reef hundreds of kilometers long in the Red Sea has been calculated to contain more material than all the buildings in the major cities of North America combined. Corals flourish in nutrient-poor, clear, tropical waters. They can grow rapidly in such environments because of photosynthetic dinoflagellates that live symbiotically within their cells, provide them with products of photosynthesis and contribute to calcium deposition. In turn, the corals provide the dinoflagellates with a place to live and nutrients. This symbiotic relationship explains why reef-forming corals are restricted to clear surface waters, where light levels are high enough to allow photosynthesis. Coral reefs throughout the world are being threatened both by global warming, which is raising the temperatures of shallow tropical ocean waters, and by polluted runoff from development on adjacent shorelines. An overabundance of nitrogen in the runoff gives an advantage to algae, which overgrow and eventually smother the corals. Hydrozoans. Life cycles are diverse among the hydrozoans, class Hydrozoa. The polyp typically dominates the life cycle, but some species have only medusae and others only polyps. Most hydrozoans are colonial. A single planula eventually gives rise to a colony of many polyps, all interconnected and sharing a continuous gastrovascular cavity. Figure 7. Within such a colony, the man of war is an example. Some polyps have tentacles with many nematocysts. They capture prey for the colony. Others lack tentacles and are unable to feed, but are specialized for the production of medusae. Still others are finger-like and defend the colony with their nematocysts. Scyphozoans. The several hundred species of cyclozoans, class Cyclozoa, are all marine. The mesoglia of the medusae is thick and firm, giving rise to their common names, jellyfish or sea jellies. The medusa, rather than the polyp, dominates the life cycle of cyclozoans. An individual medusa is male or female, releasing eggs or sperm into the open sea. The fertilized egg develops into a small planula that quickly settles on a substratum and develops into a small polyp. This polyp feeds and grows and may produce additional polyps by budding. After a period of growth, the polyp begins to bud off small medusae. Figure 8. These medusae feed, grow, and transform themselves into adult medusae, which are commonly seen during summer in harbors and bays. Ctenophores, complete guts and tentacles. Ctenophores phylum Ctenophora were the next lineage to diverge from the lineage leading to all other animals. Ctenophores, also known as comb jellies, have body plans that are superficially similar to those of cnidarians. Both have two cell layers separated by a thick, gelatinous mesoglia, and both have radial symmetry and feeding tentacles. Like cnidarians, ctenophores have low metabolic rates because they are composed primarily of inert mesoglia. Unlike cnidarians, however, ctenophores have a complete gut. Food enters through a mouth, and wastes are eliminated through two anal pores. Ctenophores have 8 comb-like rows of fused plates of cilia, called C10s. Figure 32.12. Ctenophores move by beating these cilia rather than by muscular contractions. C10phore tentacles do not have nematocysts. Rather, they are covered with cells that discharge adhesive material when they contact prey. After capturing its prey, a C10phore retracts its tentacles to bring the food to its mouth. In some species, the entire surface of the body is coated with sticky mucus that captures prey. All of the 100 known species of ctenophores eat small animals. They are common in open seas. C10phore life cycles are simple. Gametes are produced in structures called gonads, located on the walls of the gastrovascular cavity. The gametes are released into the cavity and then discharged through the mouth or the anal pores. Fertilization takes place in open seawater. In nearly all species, the fertilized egg develops directly into a miniature C10phore that gradually grows into an adult. The evolution of bilaterally symmetrical animals. The phylogenetic tree pictured in figure 1 assumes that all bilaterally symmetrical animals share a common ancestor, but it does not tell us what that ancestor looked like. To infer the form of the earliest bilitarians, zoologists use evidence from the genes, development, and structure of existing animals. An important clue is provided by the fact that the development of all bilaterally symmetrical animals is controlled by homologous Hox and homeobox genes. Regulatory genes with similar functions are unlikely to have evolved independently in several different animal lineages. Fossilized tracks from late Precambrian times provide additional clues to the nature of early bilitarians. The complexity of the movements recorded by these tracks suggests that early bilitarians had circulatory systems, systems of antagonistic muscles, and a tissue or fluid-filled body cavity, structures that are also suggested by genetic data. An early lineage split separated protozoans and deuterostomes. The next major split in the animal lineage after the divergence of the ctenophores occurred during the Cambrian period and separated two groups that have been evolving separately ever since. These two major lineages, the protozoans and the deuterostomes, dominate today's fauna. Members of both lineages are triploblastic, have 3 cell layers, bilaterally symmetrical, and cephalized. Because their skeletons and body cavities are more complex than those of the animals we have discussed so far, they are capable of more complex movements. The most important shared, derived traits that unite the protozoans are an anterior brain that surrounds the entrance to the digestive tract, a ventral nervous system consisting of a pair of fused longitudinal nerve cords, a free-floating larva with a food-collecting system consisting of compound cilia on multiciliate cells, a blastopore that becomes the mouth, spiral cleavage in some species. The major shared, derived traits that unite the deuterostomes are a dorsal nervous system. A larva, if present, that has a food-collecting system consisting of cells with a single cilium, a blastopore that becomes the anus radial cleavage. The protozoan split into two lineages. Developmental, structural, and molecular data all suggest that the protozoan soon split into two major lineages that have been evolving independently since ancient times: Lithotrochozoans and Exozoans. Lithotrochozoans, the animals we will discuss in the remainder of this chapter, grow by adding to the size of their skeletal elements. Some of them use cilia for locomotion, and many lineages have a type of free-living larva known as a trophophore. The phylogeny of Lithotrochozoans we will use in this chapter is shown in Figure 9. In contrast, Exozoans, the animals we will discuss in the next chapter, increase in size by molding their external skeletons. They move by mechanisms other than ciliary action, and they all have a common set of homeobox genes. Simple Lithotrochozoans. The simplest Lithotrochozoans, flatworms and rotifers, are small aquatic or parasitic animals. They move by rapidly beating cilia, and most have only simple organs. Flatworms move by beating cilia. Members of the phylum Platyhelminths, or flatworms, the simplest Lithotrochozoans, Figure 11, are bilaterally symmetrical, unsegmented, acolumnate animals. They lack organs for transporting oxygen to internal tissues, and they have only simple cells for excreting metabolic wastes. Their lack of transport systems dictates that each cell must be near a body surface, a requirement met by their dorsoventrally flattened body form. The digestive tract of a flatworm consists of a mouth opening into a blind sac. However, the sac is often highly branched, forming intricate patterns that increase the surface area available for the absorption of nutrients. Flatworms either feed on animal tissues, living or dead, or absorb nutrients from a host gut. Free-living flatworms glide over surfaces, powered by broad bands of cilia. This form of movement is very slow, but it is sufficient for small, scavenging animals. The flatworms that are probably most similar to the ancestral bilitarians are the Terbellarians, class Turbellaria, which are small, free-living marine and freshwater animals, a few live in moist terrestrial habitats. At one end they have a head with chemoreceptor organs, two simple eyes, and a tiny brain composed of anterior thickenings of the longitudinal nerve cords. Although the earliest flatworms were free-living, many species evolved a parasitic existence. A likely evolutionary transition was from feeding on dead organisms to feeding on the body surfaces of dying hosts to invading and consuming parts of living, healthy hosts. Most of the 25,000 species of living flatworms, including the tapeworms class Cystota and Flukes class Trematoda, Figure 10, are internal parasites. These flatworms absorb digested food from the digestive tracts of their hosts, so many of them lack digestive tracts. They inhabit the bodies of many vertebrates. Some cause serious human diseases, such as schistosomiasis. Monogenes class Monogenea are external parasites of fishes and other aquatic vertebrates. Parasites live in nutrient-rich environments where food is delivered to them, but they face other challenges. To complete their life cycle, parasites must overcome the defenses of their host. And because they die when their host dies, they must disperse their offspring to new hosts while their host is still living. The fertilized eggs of some parasitic flatworms are voided with the host species and later ingested directly by other host individuals. However, most parasitic species have complex life cycles involving one or more intermediate hosts and several larval stages. Figure 11. Such life cycles facilitate the transfer of individual parasites among hosts. Rotifers are small but structurally complex. Rotifers phylum Rotifera are bilaterally symmetrical, unsegmented, pseudocoelenate lithotrochozoans. Most rotifers are tiny, 50-500 micrometers long, smaller than some cilia protists, but they have highly developed internal organs. Figure 12. A complete gut passes from an anterior mouth to a posterior anus. The pseudosoul functions as a hydrostatic skeleton. Most rotifers propel themselves through the water by means of rapidly beating cilia rather than by muscular contraction. This type of movement is effective because rotifers are so small. The most distinctive organs of rotifers are those they use to collect and process food. A conspicuous ciliated organ called the corona surmounts the head of many species. Coordinated beating of the cilia sweeps particles of organic matter from the water into the animal's mouth and down to a complicated structure called the mastax, in which food is ground into small pieces. By contracting the muscles around the pseudosoul, a few rotifer species that prey on protists and small animals can protrude the mastax through the mouth and seize small objects with it. Males and females are found in most species, but some species have only females that produce diploid eggs without being fertilized by a male. Some rotifers are marine, but most of the 1,800 known species live in freshwater. Members of a few species rest on the surface of mosses or lichens in a desiccated, inactive state until it rains. When rain falls, they absorb water and become mobile, feeding the films of water that temporarily cover the plants. Most rotifers live no longer than 1 or 2 weeks. Lophophorates, an ancient body plan. After the platyhelminths and rotifers diverged from it, the Lithotrochozoan lineage divided into 2 branches. The descendants of those branches became the modern Lithophorites, the subject of this section, and the Spiralians, which we will discuss in the following section. About 4,850 living species of Lithophorites are known, but many times that number of species existed during the Paleozoic and Mesozoic eras. 3 Lithophorate phyla survive today: Phoronida, Brachiopoda, and Ectoprocta. Nearly all members of these phyla are marine. Only a few species of Ectoprocs live in freshwater. Lithophorate animals obtain food by filtering it from the surrounding water, a trait they share with many other protozoans. The most conspicuous feature of these animals is the lophophore, a circular or U-shaped ridge around the mouth that bears 1 or 2 rows of ciliated, hollow tentacles. Figure 13. This large and complex structure is an organ for both food collection and gas exchange. Nearly all adult Lithophorate animals are sessile, and they use the tentacles and cilia of their lophophore to capture plankton, small floating organisms, from the water. Lithophorates also have a shaped gut. The anus is located close to the mouth, but outside the tentacles. Phoronids are sedentary Lithophorates. The 20 known species of Phoronids, phylum Phoronida, are sedentary worms that live in muddy or sandy sediments or attached to a rocky substratum. Phoronids are found in marine waters ranging from intertidal zones to about 400 meters deep. They range in size from 5 to 25 centimeters in length. They secrete chitinous tubes, in which they live. Figure 13. The lophophore is the most conspicuous external feature of the Phoronids. Cilia drive water into the top of the lophophore, and water exits through the narrow spaces between the tentacles. Suspended food particles are caught and transported to the mouth by ciliary action. In most species, eggs are released into the water, where they are fertilized, but some species produce large eggs that are fertilized internally, where they are brooded until they hatch. Ectoprocs are colonial Lithophorites. Ectoprocs, phylum Ectoprocta, are colonial Lithophorites that live in a house made of material secreted by the external body wall. A colony consists of many small 1 to 2 individuals connected by strands of tissue along which materials can be moved. Most of the 4,500 species of Ectoprocs are marine, but a few live in freshwater. They are able to oscillate and rotate the lophophore to increase contact with prey. Figure 14, and can retract it into the tube. A colony of Ectoprocs is created by the asexual reproduction of its founding members. A single colony may contain as many as 2 million individuals. In some species, individual colony members are specialized for feeding, reproduction, defense, or support. Ectoprocs reproduce sexually by releasing sperm into the water, where they are collected by other individuals. Eggs are fertilized internally, and developing embryos are brooded before they exit as larvae to seek suitable sites for attachment to the substratum. Brachiopods superficially resemble bivalve mollusks. Brachiopods, phylum Brachiopoda, are solitary marine Lithophorate animals. Their shells are divided into 2 parts that are connected by ligament. The 2 halves can be pulled shut to protect the soft body. Brachiopods superficially resemble bivalve mollusks, but the brachiopod shell differs from mollusks in that the 2 halves are dorsal and ventral rather than lateral. The 2-horned lophophore of the brachiopod is located within the shell. The beating of cilia on the lophophore draws water into the slightly open shell. Food is trapped in the lophophore and directed to a ridge, along which it is transferred to the mouth. Most brachiopods are between 4 and 6 centimeters long, but some are as long as 9 centimeters. Brachiopods live attached to a solid substratum or embedded in soft sediments. Most species are attached by means of a short, flexible stalk that holds the animal above the substratum. Gases are exchanged across body surfaces, especially the tentacles of the lophophore. Most brachiopods release their gametes into the water, where they are fertilized. The larvae remain among the plankton for only a few days before they settle and develop into adults. Brachiopods reach their peak abundance and diversity in Paleozoic and Mesozoic times. More than 26,000 fossil species have been described. Only about 335 species survive, but they are common in some marine environments. Only about 335 species survive, but they are common in some marine environments. Spiralians, spiral cleavage and worm-like body plans. The Spiralian lineage, containing animals that typically have spiral cleavage patterns, gave rise to many phyla. Members of more than a dozen of these phyla are worm-like, that is, they are bilaterally symmetrical, legless, soft-bodied, and at least several times longer than they are wide. This body form enables animals to move efficiently through muddy and sandy marine sediments. Most of these phyla have no more than several hundred species. The most species-rich Spiralian phylum, the mollusks, show significant modifications of the worm-like body plan. Ribbon worms are unsegmented. The carnivorous ribbon worms, phylum Lemuria, are dorsoventrally flattened. They have nervous and excretory systems similar to those of flatworms, but unlike flatworms, they have a complete digestive tract with a mouth at one end and an anus at the other. Food moves in one direction through the digestive tract and is acted on by a series of digestive enzymes. Small ribbon worms move by beating their cilia. Larger ones employ waves of muscle contraction to move over the surface of sediments or to burrow. Movement by both of these methods is slow. Within the body of nearly all of the 900 species of ribbon worms is a fluid-filled cavity called the ring cavity, within which lies a hollow, muscular proboscis. The proboscis, which is the feeding organ, may extend much to the length of the worm. Contraction of the muscles surrounding the ring causes the proboscis to be everted explosively through an anterior opening, figure 15, without moving the rest of the animal. The proboscis of most ribbon worms is armed with a sharp stylet that pierces the prey. Paralysis causing toxins produced by the proboscis are discharged into the wound. Reproduction and development in ribbon worms is highly varied. Segmentation improves locomotion in the annelids. Segmentation allows an animal to alter the shape of its body in complex ways and to control its movements more precisely. Fossils of segmented worms are known from the Middle Cambrian. The earliest forms are thought to have been burrowing marine animals. Segmentation evolved several times among spiralians. We will discuss only one of the phyla with segmented members, the annelids. The annelids phylum Annelida are a diverse group of segmented spiralian worms, figure 16. The coelom in each segment is isolated from those in other segments. A separate nerve center called the ganglion controls each segment, and the ganglia are connected by nerve cords that coordinate their functioning. Most annelids lack a rigid, external protective covering. The body wall serves as a general surface for gas exchange in most species, but this thin, permeable body surface restricts annelids to moist environments. They lose body water rapidly in dry air. The approximately 16,500 described species live in marine, freshwater, and terrestrial environments. Polychaetes. More than half of all annelid species are members of the class Polychaeta, many hairs. Nearly all polychaetes are marine animals. Most have one or more pairs of eyes and one or more pairs of tentacles at the anterior end of the body. The body wall in most segments extends laterally as a series of thin outgrowths, called parapodia, that contain many blood vessels. The parapodia function in gas exchange, and some species use them to move. Stiff bristles called setae protrude from each parapodium, forming temporary attachments to the substratum that prevent the animal from slipping backward when its muscles contract. Typically, males and female polychaetes release gametes into the water, where the eggs are fertilized and develop into tropophore larvae, figure 17. The tropophore is a distinctive larval type found among polychaetes, mollusks, and several other marine lineages with spiral cleavage. The second half of the name, Liphotrochozoans, is derived from this larva, which is believed by many researchers to represent an evolutionary link between the annelids and the mollusks. As the polychaete tropophore develops, it forms body segments at its posterior end. Eventually, it becomes a small adult worm. Many polychaete species live in burrows in soft sediments and filter prey from the surrounding water with elaborate, feathery tentacles. OLIGOCHAETDS. More than 90% of the approximately 3,000 described species of oligochaetes, class Oligocata, live in freshwater or terrestrial habitats. Oligochaetes' few hairs, have no parapodia, eyes, or anterior tentacles, and they have relatively few setae. Earthworms, the most familiar oligochaetes, are scavengers and ingesters of soil, from which they extract food particles. Unlike polychaetes, all oligochaetes are hermaphroditic, that is, each individual is both male and female. Sperm are exchanged simultaneously between two copulating individuals. Eggs are laid in a cocoon outside the adult's body. The cocoon is shed, and when development is complete, miniature worms emerge and begin independent life. Leeches. Leeches, class Hirudinea, probably evolved from oligochaete ancestors. Most species live in freshwater or terrestrial habitats and, like oligochaetes, lack parapodia and tentacles. Like oligochaetes, leeches are hermaphroditic. The coelom of leeches is not divided into compartments. The colonic space is largely filled with indifferentiated tissue. Groups of segments at each end of the body are modified to form suckers, which serve as temporary anchors that aid the leech in movement. With its posterior sucker attached to a substratum, the leech extends its body by contracting its circular muscles. The anterior sucker is then attached, the posterior one detached, and the leech shortens itself by contracting its longitudinal muscles. Many leeches are external parasites of other animals, but some species also eat snails and other invertebrates. A leech makes an incision in its host, from which blood flows. It can ingest so much blood in a single feeding that its body may enlarge several times. An anticoagulant secreted by the leech into the wound keeps the host blood flowing. For hundreds of years, leeches were widely employed in medicine. Even today, they are used to reduce fluid pressure and prevent blood clotting in damaged tissues and to eliminate pools of coagulated blood. BESTIMENTIFERANS. Members of one lineage of annelids, the Bestimityphrans class Pogonoptera, evolved burrowing forms with a crown of tentacles through which gases are exchanged. They entirely lost their digestive systems. Bestimityphrans secrete chitin and other substances to form the tubes in which they live. A Bestimityphran coelom consists of an anterior compartment into which the tentacles can be withdrawn, and a long, subdivided cavity that extends much of the length of its body. The posterior end of the body is segmented. Experiments using radioactively labeled molecules have shown that Bestimityphrans take up dissolved organic matter at high rates from sediments in which they live or the surrounding water. Bestimityphrans were not discovered until the 20th century, when deep sea exploration revealed them living many thousands of meters below the ocean surface. In these deep oceanic sediments, they are abundant, reaching densities of many thousands per square meter. About 145 species have been described. The largest and most remarkable Bestimityphrans, which grow to 2 meters in length, live near deep sea hydrothermal vents, volcanic openings in the seafloor through which hot, sulfide-rich water pours. The tissues of these species harbor endosymbiotic bacteria that fix carbon using energy obtained from oxidation of hydrogen sulfide H2S. Mollusks evolved shells. Mollusks phylum Mollusca range in size from snails only a millimeter high to giant squids more than 18 meters long, the largest known invertebrates. Mollusks underwent one of the most dramatic of animal evolutionary radiations, based on a unique body plan with three major structural components: a foot, a mantle, and a visceral mass. Animals that appear very different, such as snails, clams, and squids, are all built from these components, figure 18. The molluscan foot is a large, muscular structure that originally was both an organ of locomotion and support for the internal organs. In the lineage leading to squids and octopuses, the foot was modified to form arms and tentacles borne on a head with complex sense organs. In other groups, such as clams, the foot was transformed into a burrowing organ. In some lineages, the foot is greatly reduced. The mantle is a fold of tissue that covers the visceral mass of internal organs. In many mollusks, the mantle extends beyond the visceral mass to form a mantle cavity. The mantle secretes a hard, calcareous skeleton typical of most mollusks. The gills, which are used for gas exchange and, in some species, for feeding, lie in this cavity. When the cilia on the gills beat, they create a flow of water over the gills. The tissue of the gills, which is highly vascularized, contains many blood vessels, takes up O2 from the water and releases CO2. Mollusks have an open circulatory system that empties into large fluid-filled cavities, through which fluids move around the animal and deliver O2 to internal organs. Mollusks also developed a rasping feeding structure known as the radula. The radula was originally an organ for scraping algae from rocks, a function it retains in many living mollusks. However, in some mollusks, it has been modified into a drill or poison dart. In others, such as clams, it is absent. Although individual components have been lost in some lineages, these three unique, shared derived characteristics, the foot, the mantle, and the visceral mass, lead zoologists to believe that all 95,000 species of mollusks have a common ancestor. MONOPLACOPHORANS. Monoplacophorans class Monoplacophora were the most abundant mollusks during the Cambrian period, 550 million years ago, but today there are only a few surviving species. Unlike all other living mollusks, the surviving monoplacophorans have respiratory organs, muscles, and excretory pores that are repeated over the length of the body. The respiratory organs are located in a large cavity under the shell, through which oxygen-bearing water circulates. Chitins. Chitins class Polyplacophora have multiple gills and shell plates, but the body is not truly segmented. The chitin body is bilaterally symmetrical, and its internal organs, particularly the digestive and nervous systems, are relatively simple. The larvae of chitins are almost indistinguishable from those of annelids. Most chitins are marine herbivores that scrape algae from rocks with their sharp gradually. An adult chitin spends most of its life clinging tightly to rock surfaces with its large, muscular, mucus-covered foot. It moves slowly by means of rippling waves of muscular contraction in the foot. Fertilization in most chitins takes place in the water, but in a few species fertilization is internal and embryos are brooded within the body. Bivalves. One lineage of early mollusks developed a hinge, tuberous shell that extended over the sides of the body as well as the top, giving rise to the bivalves class Bivalvia, which include the familiar clams, oysters, scallops, and mussels. Bivalves are largely sedentary and have greatly reduced heads. The foot is compressed, and in many clams, it is used for burrowing into mud and sand. Bivalves feed by taking in water through an opening called an inferent siphon and extracting food from the water with their large gills, which are also the main sites of gas exchange. Water and gametes exit through the exferent siphon. Fertilization takes place in open water in most species. Gastropods. Another lineage of early mollusks gave rise to the gastropods class Gastropoda, which includes snails, whelks, limpets, slugs, abalones, and the often brilliantly ornamented nudibranchs. Gastropods, unlike bivalves, have one-piece shells. Most gastropods are mobile, using the large foot to move slowly across the substratum or to burrow through it. Gastropods are the most species rich and widely distributed of the molluscan classes. Most species move by gliding on the muscular foot, but in a few species, the sea butterflies and heteropods, the foot is modified into a swimming organ with which the animal moves through open ocean waters. The only mollusks that live in terrestrial environments, land snails and slugs, are gastropods. In these terrestrial species, the mantle tissue is modified into a highly vascularized lung. Fertilization is internal in most species. Cephalopods. In one lineage of mollusks, the cephalopods class Cephalopoda, the exferent siphon became modified to allow the animal to control the water content of the mantle cavity. Ultimately, the modification of the mantle into a device for forcibly ejecting water from the cavity enabled these animals to move rapidly through the water. Furthermore, many early cephalopods had chambered shells into which gas could be secreted to adjust buoyancy. Together, these adaptations allow cephalopods to live in open water. The cephalopods include the squids, octopuses, and nautiluses. They first appeared about 600 million years ago, near the beginning of the Cambrian period. By the Ordovician period, a wide variety of types were present. With their greatly enhanced mobility, some cephalopods, such as squids, became the major predators in the open waters of the Devonian oceans. They remain important marine predators today. Cephalopods capture and subdue their prey with their tentacles. Octopuses also use their tentacles to move over the substratum. As is typical of active predators, cephalopods have a head with complex sensory organs, most notably eyes that are comparable to those of vertebrates in their ability to resolve images. The head is closely associated with a large, branched foot that bears the tentacles and a siphon. The large muscular mantle provides a solid external supporting structure. The gills hang in the mantle cavity. As is typical of behaviorally complex animals, many cephalopods have elaborate courtship behavior, which may involve striking color changes. The earliest cephalopod shells were divided by partitions penetrated by tubes through which liquids could be moved. Nautiloids genus Nautilus are the only cephalopods with external chambered shells that survive today. Mollusks and brachiopods are among the lophotrachozoans that evolved hard shells that help to protect them from predators in the physical environment. A sturdy outer covering is the main feature of the second protostomate lineage, the ichthysozoans, the subject of the next section. ECDYSOZOANS, the molting animals. Early in animal evolution, the protostomate lineage split into two branches, the lophotrachozoans and the ichthysozoans, as we saw in the previous chapter. The distinguishing feature of the ichthysozoans is an exoskeleton, an unliving covering that provides an animal with both protection and support. Once formed, however, an exoskeleton cannot grow. How, then, can ichthysozoans increase in size? Their solution is to shed, or molt, the exoskeleton and replace it with a new, larger one. Before the animal molts, a new exoskeleton is already forming underneath the old one. When the old exoskeleton is shed, the new one expands and hardens. But until it has hardened, the animal is very vulnerable to its enemies both because its outer surface is easy to penetrate and because it can move only slowly. The exoskeleton presented new challenges in other areas besides growth. Ecdysozoans cannot use cilia for locomotion, and most exozoans have hard exoskeletons that impede the passage of oxygen into the animal. To cope with these challenges, ecdysozoans evolved new mechanisms of locomotion and respiration. Despite these constraints, the ichthysozoans, the molting animals, have more species than all other animal lineages combined. An increasingly rich array of molecular and genetic evidence, including a set of homeobox genes shared by all ichthysozoans, suggests that molting may have evolved only once during animal evolution. In this chapter, we will review the diversity of the ichthysozoans. We will look at the characteristics of animals in the various ichthyzoan phyla and see how having an exoskeleton has influenced their evolution. The phylogeny we will follow is presented in figure 19. Cubicles, flexible, unsegmented exoskeletons. Some ichthyzozoans have worm-like bodies covered by exoskeletons that are relatively thin and flexible. Such an exoskeleton, called a cubicle, offers the animal some protection, but does not provide body support. The action of circular and longitudinal muscles on fluids in the body cavity provides a hydrostatic skeleton for many of these animals, which can move only slowly. A cubicle allows the exchange of gases, minerals, and water across the body surface, but restricts the animal to moist habitats. Some marine ichthyzoan phyla have few species. Several phyla of marine worm-like animals branch off early within the ichthyzoan lineage. Each of these phyla contains only a few species. These animals have relatively thin cubicles that are molted periodically as the animals grow to full size. Priapulids and kinorhynchs. The 16 species of Priapulids, phylum Priapulida, are cylindrical, unsegmented, worm-like animals that range in size from half a millimeter to 20 centimeters in length. Figure 33.2. They burrow in fine marine sediments and prey on soft-bodied invertebrates, such as filiate worms. They capture prey with a toothed pharynx, a muscular organ that is everted through the mouth and then withdrawn into the body together with the grasped prey. Fertilization is external, and most species have a larval form that lives in the mud. About 150 species of kinorhynchs, phylum Kinorhyncha, have been described. They are all less than 1 millimeter in length and live in marine sands and muds. Their bodies are divided into 13 segments, each with a separate cuticular plate. These plates are periodically molted during growth. Kinorhynchs feed by ingesting sediments and digesting the organic material found within them, which may include living algae as well as dead matter. Kinorhynchs have no distinct larval stage. Fertilized eggs develop directly into juveniles, which emerge from their egg cases with 11 of the 13 body segments already formed. Arrowworms. The phylogeny of the arrowworms, phylum Chaetognatha, is uncertain. Recent evidence indicates that these animals may in fact belong among the deuterostomes. However, this placement is still in question, and we continue to include them among the ichthysozoans. The arrowworm's body plan is based on a ceiling divided into head, trunk, and tail compartments. Figure 21. Most arrowworms swim in the open sea, but a few live on the seafloor. Their abundance of fossils indicates that they were common more than 500 million years ago. The 100 or so living species of arrowworms are small enough, less than 12 centimeters long, that their gas exchange and excretion requirements are met by diffusion through the body surface, and they lack a circulatory system. Waste and nutrients are moved around the body in the colonic fluid, which is propelled by cilia that line the coelom. There is no distinct larval stage. Miniature adults hatch directly from eggs that are fertilized internally following elaborate courtship. Arrowworms are stabilized in the water by means of one or two pairs of lateral fins and a tail fin. They are major predators of small organisms in the open oceans, ranging in size from small protists to young fish as large as the arrowworms themselves. An arrowworm typically lies motionless in the water until water movement signals the approach of prey. The arrowworm then darts forward and grasps the prey with the stiff spines adjacent to its mouth. Tough cuticles evolved in some unsegmented worms. Tough external cuticles evolved in some members of another ichthyzoan lineage whose descendants colonized freshwater and terrestrial as well as marine environments. Two extant phyla represent this lineage. Horsehair worms. About 320 species of horsehair worms, phylum Nematomorpha, have been described. As their name implies, horsehair worms are extremely thin, and they range from a few millimeters up to a meter in length. Most adult horsehair worms live in freshwater among leaf litter and algal mats near the edges of streams and ponds. The larvae of horsehair worms are internal parasites of terrestrial and aquatic insects and freshwater crayfish. The horsehair worm's gut is greatly reduced, has no mouth opening, and is probably non-functional. These worms may feed only as larvae, absorbing nutrients from their hosts across the body wall, but many continue to grow after they have left their hosts, suggesting that adult worms may also absorb nutrients from their environment. Roundworms. Roundworms, phylum Nematoda, have a thick, multi-layered cuticle secreted by the underlying epidermis that gives their body its shape. Figure 22. As a roundworm grows, it sheds its cuticle 4 times. Roundworms exchange oxygen and nutrients with their environment through both the cuticle and the intestine, which is only 1 cell layer thick. Materials are moved through the gut by rhythmic contraction of a highly muscular organ, the pharynx, at the worm's anterior end. Roundworms move by contracting their longitudinal muscles. Roundworms are one of the most abundant and universally distributed of all animal groups. About 25,000 species have been described, but the actual number of living species may be more than a million. Countless roundworms live as scavengers in the upper layers of the soil, on the bottoms of lakes and streams, in marine sediments, and as parasites in the bodies of most kinds of plants and animals. The topsoil of rich farmland contains up to 3 billion nematodes per acre. Many roundworms are predators, preying on protists and other small animals, including other roundworms. Many roundworms live parasitically within their hosts. The largest known roundworm, which reaches a length of 9 meters, is a parasite in the placentas of female sperm whales. The roundworms that are parasites of humans causing serious tropical diseases such as trichinosis, filariasis, and elephantiasis domestic animals, and economically important plants have been studied intensively in an effort to find ways of controlling them. One soil-inhabiting nematode, Canner habitus elegans, is a model organism in the laboratories of geneticists and developmental biologists. The structure of parasitic roundworms is similar to that of free-living species, but the life cycles of many parasitic species have special stages that facilitate the transfer of individuals among hosts. Trichinella spiralis, the species that causes the human disease trichinosis, has a relatively simple life cycle. A person may become infected by eating the flesh of an animal, usually a pig containing larvae of trichinella encysted in its muscles. The larvae are activated in the digestive tract, emerge from their cysts, and attach to the person's intestinal wall, where they feed. Later, they bore through the intestinal wall and are carried in the bloodstream to muscles, where they form new cysts. If present in great numbers, these cysts cause severe pain or death. Arthropods and their relatives, segmented external skeletons. In Precambrian times, the cuticle of some worm-like ichthyzoan lineages became thickened by the incorporation of layers of protein and a strong, flexible, waterproof polysaccharide called chitin. This rigid body covering may originally have had a protective function, but eventually it acquired both support and locomotory functions as well. A rigid body covering precludes worm-like movement. To move, the animal requires extensions of the body that can be manipulated by muscles. Such appendages evolved several times in the late Precambrian, leading to the lineages collectively called the arthropods jointed foot. Divisions among the arthropod lineages are ancient and have been the subject of much research in the past decade. These phylogenetic relationships are being examined daily in the light of a wealth of new information, much of it concerning gene expression. There is currently no consensus on an exact phylogeny, but most researchers agree that these important animal groups are monophyletic, and some taxonomists consider them as members of a single phylum, Arthropoda. Before presenting one current view of arthropod phylogeny, let's look at some arthropod relatives that have segmented bodies but unjointed legs, and an early arthropod lineage that disappeared but left an important fossil record. Some relatives of the arthropods have unjointed legs. Although they were once thought to be closely related to annelid worms, recent molecular evidence links the 110 species of onychophorans phylum Onychophora to the arthropod lineages. Onychophorans have soft bodies that are covered by a thin, flexible cuticle that contains chitin. Onychophorans use their fluid-filled body cavities as hydrostatic skeletons. Their soft, fleshy, unjointed, claw-bearing legs are formed by outgrowths of the body. These animals are probably similar in appearance to ancestral arthropods. Fertilization is internal, and the large, yolky eggs are brooded with the body of the female. Like the onychophorans, water bears phylum Tardigrada have fleshy, unjointed legs and use their fluid-filled body cavities as hydrostatic skeletons. Water bears are extremely small, 0.1-0.5 millimeters in length, and they lack circulatory systems and gas exchange organs. The 600 extant species of water bears live in marine sands and on temporary water films on plants. When these films dry out, the water bears also lose water and shrink to small, barrel-shaped objects that can survive for at least a decade in a dormant state. They have been found at densities as high as 2 million per square meter of moss. Jointed legs appeared in the trilobites. The trilobites phylum Trilobita were among the earliest arthropods. They flourished in Cambrian and Ordovician seas, but disappeared in the Great Permian extinction at the close of the Paleozoic era 245 Maya. Because their heavy exoskeletons provided ideal material for fossilization, they left behind an abundant record of their existence. Trilobites were heavily armored, and their body segmentation and appendages followed a relatively simple, repetitive plan. But their appendages were jointed, and some of them were modified for different functions. This specialization of appendage function became a theme as the evolution of the arthropod lineage continued. Modern arthropods dominate Earth's fauna. Arthropod appendages have evolved in an amazing variety of forms, and they serve many functions, including walking and swimming, gas exchange, food capture and manipulation, copulation, and sensory perception. The pattern of segmentation is similar among most arthropods because their development is governed by a common cascade of regulatory genes, including homeotic genes that determine the kinds of appendages that are born on each segment. The bodies of arthropods are divided into segments. Their muscles are attached to the inside of the exoskeleton. Each segment has muscles that operate the segment and the appendage is attached to it. Figure 23. The arthropod exoskeleton has had a profound influence on the evolution of these animals. Encasement within a rigid body covering provides support for walking on dry land, and the waterproofing provided by chitin keeps the animal from dehydrating in dry air. Aquatic arthropods were, in short, excellent candidates to invade terrestrial environments. As we will see, they did this several times. There are 4 major arthropod phyla living today: the crustaceans, hexapods, insects, myriapods, and chelicerates. Collectively, the arthropods, including both terrestrial and marine species, are the dominant animals on Earth, both in numbers of species, about 1.5 million described, and number of individuals estimated at some 10-18 individuals, or a billion. Crustaceans, diverse and abundant. Crustaceans phylum Crustacea are the dominant marine arthropods today. The most familiar crustaceans belong to the class Molluscostraca, which includes shrimp, lobsters, crayfish, and crabs, decapods, and sobugs, isopods. Also included among the crustaceans are a variety of small species, many of which superficially resemble shrimp. The individuals of one group alone, the copods class Copepoda, are so numerous that they may be the most abundant of all animals. Barnacles class Cirripedia are unusual crustaceans that are sessile as adults. With their calcareous shells, they superficially resemble mollusks, but, as the zoologist Louis Agassiz remarked more than a century ago, a barnacle is nothing more than a little shrimp-like animal, standing on its head in a limestone house and kicking food into its mouth. Most of the 40,000 described species of crustaceans have a body that is divided into 3 regions: head, thorax, and abdomen. The segments of the head are fused together, and the head bears 5 pairs of appendages. Each of the multiple thoracic and abdominal segments usually bears 1 pair of appendages. In some cases, the appendages are branched, with different branches serving different functions. In many species, a fold of the exoskeleton, the carapace, extends dorsally and laterally back from the head to cover and protect some of the other segments. Figure 24a. The fertilized eggs of most crustacean species are attached to the outside of the female's body, where they remain during their early development. At hatching, the young of some species are released as larvae. Those of other species are released as juveniles that are similar in form to the adults. Still other species release eggs into the water or attach them to an object in the environment. The typical crustacean larva, called a nauplius, has 3 pairs of appendages and 1 central eye. Figure 24b. In many crustaceans, the nauplius larva develops within the egg before it hatches. There is a growing recognition among researchers that a crustacean lineage may have been ancestral to all present-day arthropods. Therefore, the phylum Crustacea, as we recognize it here, may be paraphyletic. Molecular evidence points especially to a link between the crustaceans and another important lineage, the hexapods. Insects, terrestrial descendants of marine crustaceans. During the Devonian, more than 400 million years ago, arthropods made the leap from the marine environment onto land. Of the several groups who successfully colonized the terrestrial habitat, none is more prominent today than the 6-legged individuals of the phylum Hexapoda, the insects. Insects are found in most terrestrial and freshwater habitats, and they utilize nearly all species of plants and many species of animals as food. Some are internal parasites of plants and animals, others suck their host blood or consume surface body tissues. The 1.4 million species of insects that have been described are believed to be only a small fraction of the total number of species living today. Very few insect species live in the ocean. In freshwater environments, on the other hand, they are sometimes the dominant animals, burrowing through the substratum, extracting suspended prey from the water, and actively pursuing other animals. Insects were the first animals to achieve the ability to fly, and they are important pollinators of flowering plants. Insects, like crustaceans, have 3 basic body regions: head, thorax, and abdomen. They have a single pair of antennae on the head and 3 pairs of legs attached to the thorax. Figure 25. Unlike the other arthropods, insects have no appendages growing from their abdominal segments. An insect exchanges gases by means of air sacs and tubular channels called tracheae, singular, trachea, that extend from external openings inward to tissues throughout the body. The adults of most flying insects have 2 pairs of stiff, membranous wings attached to the thorax. However, flies have only 1 pair of wings, and in beetles the 4 wings form heavy, hardened wing covers. Wingless insects include springtails and silverfish. Of the modern insects, they are probably the most similar in form to insect ancestors. Apterogoat insects have a simple life cycle, hatching from eggs as miniature adults. Development in the winged insects is complex. The hatchlings do not look like adults, and they undergo substantial changes at each molt. The immature stages of insects between molts are called instars. A substantial change that occurs between one developmental stage and another is called metamorphosis. If the changes between its instars are gradual, an insect is said to have incomplete metamorphosis. In some insect groups, the larval and adult forms appear to be completely different animals. The most familiar example of such complete metamorphosis occurs in members of the order Lepidoptera, in which the larval caterpillar transforms itself into the adult butterfly. During complete metamorphosis, the worm-like larva transforms itself during a specialized phase, called the pupa, in which many larval tissues are broken down and the adult form develops. In many of these groups, the different life stages are specialized for living in different environments and using different food sources. In many species, the larvae are adapted for feeding and growing, and the adults are specialized for reproduction and dispersal. Entomologists divide the winged insects into about 29 different orders. We can make sense of this bewildering variety by recognizing 3 major lineages. Winged insects that cannot fold their wings against the body. Winged insects that can fold their wings and that undergo incomplete metamorphosis. Winged insects that can fold their wings and that undergo complete metamorphosis. We can make sense of this bewildering variety by recognizing 3 major lineages. Winged insects that cannot fold their wings against the body. Winged insects that can fold their wings and that undergo incomplete metamorphosis. Winged insects that can fold their wings and that undergo complete metamorphosis. Because they can fold their wings over their backs, flying insects belonging to the 2nd and 3rd lineages can tuck their wings out of the way upon landing and crawl into crevices and other tight places. The only surviving members of the lineage whose members cannot fold their wings against the body are the orders Odonata, dragonflies and damselflies, and Ephemeroptera mayflies. All members of these 2 orders have aquatic larvae that transform themselves into flying adults after they crawl out of the water. Although many of these insects are excellent flyers, they require a great deal of open space in which to maneuver. Dragonflies and damselflies are active predators as adults, but adult mayflies lack functional digestive tracts and live only long enough to mate and lay eggs. The 2nd lineage, whose members can fold their wings and have incomplete metamorphosis, includes the orders Orthoptera, grasshoppers, crickets, roaches, mantids, and walking sticks, Isoptera, termites, Plecoptera, stoneflies, Dermaptera, earwigs, Thysanoptera, thrips, Hemiptera, true bugs, and Homoptera, aphids, cicadas, and leafhoppers. In these groups, hatchlings are sufficiently similar in form to adults to be recognizable. They acquire adult organ systems, such as wings and compound eyes, gradually through several juvenile instars. Remarkably, a new insect order in this lineage, the Mantiphasmatodea, was first described in 2002. These small insects are common in the Cape region of southern Africa, an area of exceptional species richness and endemism for many animal and plant groups. Insects belonging to the 3rd lineage undergo complete metamorphosis. About 85% of all species of winged insects belong to this lineage. Familiar examples are the orders Neuroptera, lacewings and their relatives Coleoptera, beetles, Trichoptera, caddisflies, Lepidoptera, butterflies, and moths, Diptera, flies, and Hymenoptera, sawflies, bees, wasps, and ants. Members of several orders of winged insects, including the Thyroptera, lice, and Psychonoptera, fleas, are parasitic. Although descended from flying ancestors, these insects have lost the ability to fly. Molecular data suggests that the lineage leading to the insects separated from the lineage leading to modern crustaceans about 450 million years ago, about the time of the appearance of the first land plants. These ancestral forms penetrated a terrestrial environment that was ecologically empty, which in part accounts for their remarkable success. But this success of the insects is also due to their wings, which arose only once early during insect evolution. Homologous genes control the development of insect wings and crustacean appendages, suggesting that the insect wing evolved from a dorsal branch of a crustacean limb, figure 26. The dorsal limb branch of crustaceans is used for respiration and osmoregulation. This finding suggests that the insect wing evolved from a gill-like structure that had a respiratory function. Arthropods with two body regions. Insects and most crustaceans have tripartite body plans, with a head, thorax, and abdomen. In two other arthropod lineages, evolution resulted in a body plan with two regions, a head and a trunk. Myriapods have many legs. Centipedes, millipedes, and the two other groups of animals comprise the phylum Myriapoda. Centipedes and millipedes have a well-formed head and a long, flexible, segmented trunk that bears many pairs of legs. Centipedes, which have one pair of legs per segment, prey on insects and other small animals. Millipedes, which have two pairs of legs per segment, scavenge and eat plants. More than 3,000 species of centipedes and 10,000 species of millipedes have been described. Many more species probably remain unknown. Although most myriapods are less than a few centimeters long, some tropical species are 10 times that size. Most chelicerates have 4 pairs of walking legs. In the body plan of chelicerates, phylum Cheliceratta, the anterior region head bears 2 pairs of appendages modified to form mouthparts. In addition, many chelicerates have 4 pairs of walking legs. The 63,000 described chelicerate species are usually placed in 3 classes: Pycnogonida, Merostomata, and Arachnida. Most of them belong to Arachnida. The Pycnogonids class Pycnogonida, or sea spiders, are a poorly known group of about 1,000 marine species. Most are small, with legs. Most are small, with leg spans less than 1 centimeter, but some deep sea species have leg spans up to 60 centimeters. A few Pycnogonids feed on algae, but most are carnivorous, feeding on a variety of small invertebrates. The class Merostomata contains the horseshoe crabs, Order Zipposera, with 5 living species, and the extinct giant water scorpions, Order Eurypterida. Horseshoe crabs, which have changed very little during their long fossil history, have a large horseshoe-shaped covering over most of the body. They are common in shallow waters along the eastern coast of North America and Southeast Asia, where they scavenge and prey on bottom-dwelling invertebrates. Periodically they crawl into the intertidal zone in large numbers to mate and lay eggs. Arachnids class Arachnida are abundant in terrestrial environments. Most arachnids have a simple life cycle in which miniature adults hatch from internally fertilized eggs and begin independent lives almost immediately. Some arachnids retain their eggs during development and give birth to live young. The most species-rich and abundant arachnids are the spiders, scorpions, harvestmen, mites, and ticks. The 30,000 described species of mites and ticks live in soil, leaf litter, mosses, and lichens, underbark, and as parasites of plants, invertebrates, and vertebrates. They are vectors for weed and rhinosaic viruses, and they cause mange in domestic animals and skin irritation in humans. Spiders are important terrestrial predators. Some have excellent vision that enables them to chase and seize their prey. Others spin elaborate webs made of protein threads in which they snare prey. The threads are produced by modified abdominal appendages connected to internal glands that secrete the proteins, which dry on contact with air. The webs of different groups of spiders are strikingly varied, and this variation enables the spiders to position their snares in many different environments. Spiders also use protein threads to construct safety lines during climbing in its homes, mating structures, protection for developing young, and means of dispersal. Themes in the evolution of protozoans. We end this section by reviewing some of the evolutionary trends we have seen in the animal groups we have discussed so far. Most of protosomate evolution took place in the oceans. Early protozoans used their fluid-filled body cavities as hydrostatic skeletons. Segmentation permitted different parts of the body to be moved independently of one another. Thus, species in some protosomate lineages gradually evolved the ability to change their shape in complex ways and to move rapidly over and through the substratum or through the water. During much of animal evolution, the only food in the water consisted of dissolved organic matter and very small organisms. Consequently, many different lineages of animals, including lithophrates, mollusks, tunicates, and some crustaceans, evolved feeding structures designed to filter small prey from water, as well as structures for moving water through or over their pre-collecting devices. Animals that feed in this manner are abundant and widespread in marine waters today. Because water flows readily, bringing food with it, sessile lifestyles also evolved repeatedly during Lithotrochozoan and Ixozoan evolution. Most phyla today have at least some sessile members. Being sessile presents certain challenges. For example, sessile animals cannot come together to mate. Some species eject both eggs and sperm into the water. Others retain their eggs within their bodies and extrude only their sperm, which are carried by the water to other individuals. Species whose adults are sessile often have motile larvae, many of which have complicated mechanisms for locating suitable sites on which to settle. A sessile animal gains access to local resources, but forfeits access to more distant resources. Many colonial sessile protozoans, however, are able to grow in the direction of better resources or into sites offering better protection. Individual members of colonies, if they are directly connected, can share resources. The ability to share resources enables some individuals to specialize for particular functions, such as reproduction, defense, or feeding. The non-feeding individuals derive their nutrition from their feeding associates. Predation may have been the major selective pressure for the development of hard, external body coverings. Such coverings evolved independently in many Lithotrochozoan and Ixozoan lineages. In addition to providing protection, they became key elements in the development of new systems of locomotion. Locomotory abilities permitted prey to escape more readily from predators, but also allowed predators to pursue their prey more effectively. Thus, the evolution of animals has been, and continues to be, a complex, arms race among predators and prey. Although we have concentrated on the evolution of greater complexity in animal lineages, many lineages whose members have remained simple have been very successful. Cnidarians are common in the oceans, roundworms are abundant in most aquatic and terrestrial environments. Parasites have lost complex body plans but have evolved complex life cycles. The characteristics of the major existing phyla of protostomate animals are summarized in Table 1. Many major evolutionary trends were shared by protozoans and deuterostomes, the lineage that includes the chordates, the group to which humans belong. We will consider the evolution of diversity among the deuterostomes in the next section. Deuterostomata animals. Complex social systems, in which individuals associate with one another to breed and care for their offspring, characterize many species of fish, birds, and mammals, the most conspicuous and familiar deuterostomate animals. We tend to think of these social systems as having evolved relatively recently, but some amphibians, members of an ancient deuterostomate group, also have elaborate courtship and parental care behavior. For example, the male of the European midwife toad gathers eggs around his hind legs as the female lays them. He then carries the eggs until they are ready to hatch. In the Saurian toad, mating and parental care are exquisitely coordinated, as an elaborate mating dance results in the female depositing eggs on the male's belly. The male fertilizes the eggs and, as the ritual ends, he presses them against the female's back, where they are carried until they hatch. The female poison dart frog lays clutches of eggs on a leaf or on the ground, which both parents then work to keep moist and protected. When the tadpoles hatch, they wiggle onto the back of one of their parents, who then carries the tadpoles to water. There are fewer major lineages and many fewer species of deuterostomes in the protozoans Table 2, but we have a special interest in the deuterostomes because we are members of that lineage. In this chapter, we will describe and discuss the deuterostomate phyla, Echinodermata, Hemichordata, and Chordata. We close with a brief overview of some major themes in the evolution of animals. Deuterostome ancestors. A group of extinct animals known as the Ionozoans are the likely ancestors of all deuterostomes. Many fossils of these animals have been discovered in China's Yunnan province. These well-preserved fossils show that the animals had a large mouth, 6 pairs of external gills, and a lightly cuticularized, segmented posterior body section. Later in deuterostome evolution, gills became internal and were connected to the exterior via slits in the body wall. These gill slits subsequently were lost in the lineage leading to the modern echinoderms. Modern deuterostomes fall into 2 major clades, figure 27. One clade, composed of echinoderms and hemichordates, is characterized by a 3-part coelom and a bilaterally symmetrical, ciliated larva. The ancestors of the other clade, containing the chordates, had a distinctly different, non-feeding, tadpole-like larva and a unique dorsal supporting structure. Echinoderms, Pentaradal symmetry. During the evolution of one deuterostomate lineage, the echinoderms phylum Echinodermata, 2 major structural features arose. One was a system of calcified internal plates covered by thin layers of skin and some muscles. The calcified plates of early echinoderms later became enlarged and thickened until they fused inside the entire body, giving rise to an internal skeleton. The other feature was a water vascular system, a network of water-filled canals leading to extensions called tube feet. This system functions in gas exchange, locomotion, and feeding, figure 28a. Seawater enters the system through a perforated metaporite. A calcified canal leads from the metaporite to another canal that rings the esophagus, the tube leading from the mouth to the stomach. Other canals radiate from this ring canal, extending through the arms in species that have arms and connecting with the tube feet. The development of these 2 structural innovations resulted in a striking evolutionary radiation. About 23 classes of echinoderms, of which only 6 survive today, have been described from fossils. The 13,000 species described from their fossil remains are probably only a small fraction of those that actually lived. Nearly all 7,000 species that survive today live only in marine environments. Some have bilaterally symmetrical, ciliated larvae, figure 28b, that feed for some time as planktonic organisms before settling and transforming into adults with pendradial symmetry and fiber multiples of 5. Living echinoderms are members of 2 lineages, subphylum Helminthozoa and subphylum Eleutherozoa. These 2 groups differ in the form of their water vascular systems. Helminthozoans have jointed arms. Sea lilies and feather stars, class Chronoidea, are the only surviving helminthozoans. Sea lilies were abundant 300 to 500 million years ago, but only about 80 species survive today. Most sea lilies attach to a substratum by means of a flexible stalk consisting of a stack of calcareous discs. The main body of the animal is a cup-shaped structure that contains a tubular digestive system. 5 to several hundred arms, usually in multiples of 5, extend outward from the cup. The jointed calcareous plates of the arms enable them to bend. A sea lily feeds by orienting its arms in passing water currents. Food particles strike and stick to the tube feet, which are covered with mucus-secreting glands. The tube feet transfer these particles to grooves in the arms, where ciliary action carries the food to the mouth. The tube feet of sea lilies are also used for gas exchange and elimination of nitrogenous wastes. Feather stars are similar to sea lilies, but they have flexible appendages with which they grasp the substratum. Feather stars feed in much the same manner as sea lilies. They can walk on the tips of their arms or swim by rhythmically beating their arms. About 600 living species of feather stars have been described. Eleutherozoans are the dominant echinoderms. Most surviving echinoderms are members of the Eleutherozoan lineage. Biochemical data suggests that the Eleutherozoa split into two lineages, one leading to sea urchins, sand dollars, and the sea cucumbers, and the second leading to sea stars and brittle stars. Sea urchins and sand dollars, class Echinoidea, lack arms, but they share a 5-part body plan with all other echinoderms. Sea urchins are hemispherical animals that are covered with spines attached to the underlying skeleton via ball and socket joints. The spines of sea urchins come in varied sizes and shapes. A few produce toxic substances. Many sea urchins consume algae, which they scrape from rocks with a complex rasping structure. Others feed on small organic debris that they collect with their tube feet or spines. Sand dollars, which are flat and disc-shaped, feed on algae and fragments of organic matter found on the seafloor or suspended organic material. The sea cucumbers, class Halothoroidea, lack arms, and their bodies are oriented in an atypical manner for an echinoderm. The mouth is anterior and the anus is posterior, not oral and abrolate as in other echinoderms. Sea cucumbers use their tube feet primarily for attaching to the substratum rather than for moving. The anterior tube feet are modified into large, feathery, sticky tentacles that can be protruded from the mouth. Periodically, the sea cucumber withdraws the tentacles, wipes off the material that has adhered to them, and digests it. Sea stars, class Asteroidea, are the most familiar echinoderms. Their digestive organs and gonads are located in the arms. Their tube feet serve as organs of locomotion, gas exchange, and attachment. Each tube foot of a sea star is also an adhesive organ consisting of an internal ampulla connected by muscular tube to an external suction cup. The tube foot is moved by expansion and contraction of the circular and longitudinal muscles of the tube. Many sea stars prey on polychaetes, gastropods, bivalves, and fish. They are important predators in many echinoderms, PENTARADIAL symmetry. During the evolution of 1 to grossonate lineage, the echinoderms phylum Echinodermata, two major structural features arose. One was a system of calcified internal plates covered by thin layers of skin and some muscles. The calcified plates of early echinoderms later became enlarged and thickened until they fused inside the entire body, giving rise to an internal skeleton. The other feature was a water vascular system, a network of water-filled canals leading to extensions called tube feet. This system functions in gas exchange, locomotion, and feeding. Figure 28a. Seawater enters the system through a perforated metaporite. A calcified canal leads from the metaporite to another canal that rings the esophagus, the tube leading from the mouth to the stomach. Other canals radiate from this ring canal, extending through the arms in species that have arms and connecting with the tube feet. The development of these two structural innovations resulted in a striking evolutionary radiation. About 23 classes of echinoderms, of which only 6 survive today, have been described from fossils. The 13,000 species described from their fossil remains are probably only a small fraction of those that actually lived. Nearly all 7,000 species that survive today live only in marine environments. Some have bilaterally symmetrical, ciliated larvae, figure 28b, that feed for some time as planktonic organisms before settling and transforming into adults with chondrial symmetry, symmetry and fiber multiples of 5. Living echinoderms are members of two lineages, subphylum Helminthozoa and subphylum Eleutherozoa. These two groups differ in the form of their water vascular systems. Helminthozoans have jointed arms. Sea lilies and feather stars, class Chronoidea, are the only surviving helminthozoans. Sea lilies were abundant 300 to 500 million years ago, but only about 80 species survive today. Most sea lilies attach to a substratum by means of a flexible stalk consisting of a stack of calcareous discs. The main body of the animal is a cup-shaped structure that contains a tubular digestive system. 5 to several hundred arms, usually in multiples of 5, extend outward from the cup. The jointed calcareous plates of the arms enable them to bend. A sea lily feeds by orienting its arms in passing water currents. Food particles strike and stick to the tube feet, which are covered with mucus-secreting glands. The tube feet transfer these particles to grooves in the arms, where ciliary action carries the food to the mouth. The tube feet of sea lilies are also used for gas exchange and elimination of nitrogenous wastes. Feather stars are similar to sea lilies, but they have flexible appendages with which they grasp the substratum. Feather stars feed in much the same manner as sea lilies. They can walk on the tips of their arms or swim by rhythmically beating their arms. About 600 living species of feather stars have been described. Eleutherozoans are the dominant echinoderms. Most surviving echinoderms are members of the Eleutherozoan lineage. Biochemical data suggests that the Eleutherozoa split into two lineages, one leading to sea urchins, sand dollars, and sea cucumbers, and the second leading to sea stars and brittle stars. Sea urchins and sand dollars, class Echinoidea, lack arms, but they share a 5-part body plan with all other echinoderms. Sea urchins are hemispherical animals that are covered with spines attached to the underlying skeleton via ball and socket joints. The spines of sea urchins come in varied sizes and shapes. A few produce toxic substances. Many sea urchins consume algae, which they scrape from rocks with a complex rasping structure. Others feed on small organic debris that they collect with their tube feet or spines. Sand dollars, which are flat and disc-shaped, feed on algae and fragments of organic matter found on the seafloor or suspended organic material. The sea cucumbers, class Halothoroidea, lack arms, and their bodies are oriented in an atypical manner for an echinoderm. The mouth is anterior and the anus is posterior, not oral and abrolate as in other echinoderms. Sea cucumbers use their tube feet primarily for attaching to the substratum rather than for moving. The anterior tube feet are modified into large, feathery, sticky tentacles that can be protruded from the mouth. Periodically, the sea cucumber withdraws the tentacles, wipes off the material that has adhered to them, and digests it. Sea stars, class Asteroidea, are the most familiar echinoderms. Their digestive organs and gonads are located in the arms. Their tube feet serve as organs of locomotion, gas exchange, and attachment. Each tube foot of a sea star is also an adhesive organ consisting of an internal ampulla connected by muscular tube to an external suction cup. The tube foot is moved by expansion and contraction of the circular and longitudinal muscles of the tube. Many sea stars prey on polychaetes, gastropods, bivalves, and fish. They are important predators in many marine environments, such as coral reefs and rocky intertidal zones. With hundreds of tube feet acting simultaneously, a sea star can exert an enormous and continuous force. It can grasp a clam in its arms, anchor the arms with its tube feet, and, by steady contraction of the muscles in the arms, gradually exhaust the muscles the clam uses to keep its shell closed. Sea stars that feed on bivalves are able to push the stomach out through the mouth and then through the narrow space between the two halves of the bivalve shell. The stomach secretes enzymes that digest the prey. Brittle stars, class Ophiuroidea, are similar in structure to sea stars, but their flexible arms are composed of jointed hard plates. Brittle stars generally have 5 arms, but each arm may branch a number of times. Most of the 2,000 species of brittle stars ingest particles from the upper regions of sediments and assimilate the organic material from them, but some species remove suspended food particles from the water, others capture small animals. Brittle stars eject the indigestible particles through their mouths because, unlike most other echinoderms, they have only one opening to the digestive tract. An additional group, the sea daisies, class Concentricycloidea, were discovered only in 1986, and little is known about them. They have tiny disc-shaped bodies with a ring of marginal spines and 2 ring canals, but no arms. Sea daisies are found on rotting wood in ocean waters. They apparently eat prokaryotes, which they digest outside their bodies and absorb either through a membrane that covers the oral surface or via a shallow, sac-like stomach. Recent molecular data suggests that they are greatly modified sea stars. HEMICHORDATES, conservative evolution. Acorn worms and pteroranx, phylum Hemichordata, are probably similar in form to the ancestor they share with the echinoderms. They have a 3-part body plan, consisting of a proboscis, a collar, and a trunk. The 70 species of acorn worms range up to 2 meters in length. They live in burrows in muddy and sandy marine sediments. The large proboscis of an acorn worm is a digging organ, figure 29a. It is coated with a sticky mucus that traps small organisms in the sediment. The mucus and its attached prey are conveyed by cilia to the mouth. In the esophagus, the food-laden mucus is compacted into a rope-like mass that is moved through the digestive tract by ciliary action. Behind the mouth is a muscular pharynx, a tube that connects the mouth to the intestine. The pharynx opens to the outside through a number of pharyngeal slits through which water connects it. Highly vascularized tissue surrounding the pharyngeal slits serves as a gas exchange apparatus. An acorn worm breathes by pumping water into its mouth and out through its pharyngeal slits. The 10 living species of pterodranks are sedentary animals up to 12 millimeters in length that live in a tube secreted by the proboscis. Some species are solitary, others form colonies of individuals joined together. Behind the proboscis is a collar with 1 to 9 pairs of arms, bearing long tentacles that capture prey and function in gas exchange. Figure 29b. Chordates, new ways of feeding. Members of the second major lineage of deuterostomes evolved several modifications of the coelom that provided new ways of capturing and handling food. They evolved a strikingly different body plan, characterized by an internal dorsal supporting structure. The pharyngeal slits, which originally functioned as sites for the uptake of O2 and elimination of CO2, and for eliminating water, were further enlarged. The result was a phylum Chordata, a bilaterally symmetrical animals with body plans characterized by the following shared features at some stage in their development. Pharyngeal slits. A dorsal, hollow nerve cord. A ventral heart. A tail that extends beyond the anus, a dorsal supporting rod, the notochord. The notochord is a distinctive derived trait of the lineage. It is composed of a core of large cells with turgid fluid-filled vacuoles that make it rigid but flexible. In some Urochordates, the notochord is lost during metamorphosis to the adult stage. In vertebrates, it is replaced by other skeletal structures that provide support for the body. The tunicate subphylum Urochordata may be similar to the ancestors of the chordates. All 2,500 species of tunicates are marine animals, most of which are sessile as adults. Their swimming, tadpole-like larvae reveal the close evolutionary relationship between tunicates and chordates, as Darwin realized. In addition to its pharyngeal slits, a tunicate larva has a dorsal, hollow nerve cord and a notochord that is restricted to the tail region. Bands of muscles surround the notochord, providing support for the body. After a short time swimming in the water, the larva of most species settle to the seafloor and transform into sessile adults. The tunicate pharynx is enlarged into a pharyngeal basket, with which the animal feeds by extracting plankton from the water. Some Urochordates are solitary, but others produce colonies by asexual budding from a single founder. There are 3 major Urochordate groups: ascidians, ciliations, and larvaceans. More than 90% of the known species of tunicates are ascidians sea squirts. Individual sea squirts range in size from less than 1 millimeter to 60 centimeters in length, but colonies may measure several meters across. The bag-like body of an adult ascidian is enclosed in a tough tunic that is secreted by epidermal cells. The tunic is composed of proteins and a complex polysaccharide. Much of the body is occupied by a large pharyngeal basket lined with cilia, whose feeding moves water through the animal. Ciliations, salps, and others float in tropical and subtropical oceans at all depths down to 1,500 meters. They live singular in chain-like colonies up to several meters long. Larvaceans are solitary planktonic animals usually less than 5 millimeters long. They retain their notochords and nerve cords throughout their lives. The 25 species of lancelets, subphylum Cephalochordata, are small, fish-like animals that rarely exceed 5 centimeters in length. Their notochord extends the entire length of the body throughout their lives. Lancelets live partly buried in soft marine sediments. They extract small prey from the water with their pharyngeal baskets. Figure 30. A jointed vertebral column replaced the notochord in vertebrates. In another chordate lineage, the enlarged pharyngeal basket came to be used to extract prey from mud. This lineage gave rise to the vertebrates, subphylum Vertebrata. Figure 31. Vertebrates take their name from the jointed, dorsal vertebral column that replaced the notochord as their primary support. The vertebrate body plan. Figure 32 can be characterized as follows. A rigid internal skeleton, with the vertebral column as its anchor, that provides support and mobility. 2 pairs of appendages attached to the vertebral column. An anterior skull with a large brain. Internal organs suspended in a large coelom. A well-developed circulatory system, driven by contractions of the ventral heart. The ancestral vertebrates lacked jaws. They probably swam over the bottom, sucking up mud and straining it through the pharyngeal basket to extract microscopic food particles. The vascularized tissues of the basket also served a gas exchange function. These animals gave rise to the jawless fishes. One group of jawless fishes, called ostracoderms shell-skinned, evolved a bony external armor that protected them from predators. With their heavy armor, these small fish could safely swim slowly above the substratum, which was easier than having to burrow through it, as all previous sediment feeders had done. Jawless fishes could attach to dead organisms and use suction created by the pharynx to pull fluids and partly decomposed tissues into the mouth. Hagfishes and lampreys, the only jawless fishes to survive beyond the Devonian, feed on both dead and living organisms in this way. These fishes, often placed in the class Agnatha, have tough skins instead of external armor. They lack paired appendages, fins. The round mouth is a sucking organ with which the animals attach to the prey and rasp at the flesh. The lineages leading to modern hagfishes probably diverged from other groups first. See figure 31. The 60 species of hagfishes are entirely marine, but the 50 species of lampreys live in both fresh and salt water. Jaws improved feeding efficiency. During the Devonian period, many new kinds of fishes evolved in the seas, estuaries, and freshwaters. Although most of these fishes were jawless, in one lineage, some of the skeletal arches that supported the gills evolved into jaws. Figure 32. A fish with jaws can grasp and subdue large prey. Further development of jaws and teeth enabled some fishes to chew both soft and hard body parts of prey. Chewing aided chemical digestion and improved the fish's ability to extract nutrients from their prey. The dominant early jawed fishes were the heavily armored placoderms, class Placodermi. Some of these fishes evolved elaborate fins and relatively sleek body forms that improved their ability to maneuver in open water. A few became huge 10 meters long and, together with squids, cephalopod mollusks, were probably the major predators in the Devonian oceans. Despite their early abundance, however, most placoderms had disappeared by the end of the Devonian period. None survived to the end of the Paleozoic era. Fins improved mobility. Several other groups of fishes became abundant during the Devonian period. Cartilaginous fishes, class Chondrichthyes, the sharks, skates and rays, and chimeras, have a skeleton composed entirely of a firm but pliable material called cartilage. Their skin is flexible and leathery, sometimes bearing scales that give it the consistency of sandpaper. Cartilaginous fishes control their movement with pairs of unjointed appendages called fins, a pair of pectoral fins just behind the gill slits and a pair of pelvic fins just in front of the anal region. A dorsal median fin stabilizes the fish as it moves. Sharks move forward by means of lateral undulations of their bodies and tail fins. Skates and rays propel themselves by means of vertical undulating movements of their greatly enlarged pectoral fins. Most sharks are predators, but some feed by straining plankton from the water. The world's largest fish, the whale shark Rhincoon typhus, is a filter feeder. It may grow to more than 12 meters in length and weigh more than 12,000 kilograms. Most skates and rays live on the ocean floor, where they feed on mollusks and other invertebrates buried in the sediments. Nearly all cartilaginous fishes live in the oceans, but a few are estuarine or migrate into lakes and rivers. One group of stingrays is found only in river systems of South America. The chimeras are found in deep ocean waters and are seen less often than the sharks and rays. Swim bladders allow control of buoyancy. Rayfin fishes, class Actinopterygii, have internal skeletons of calcified, rigid bone rather than flexible cartilage. The outer surface of most species of rayfin fishes is covered with flat, thin, lightweight scales that provide some protection or enhance their movement through the water. The gills of rayfin fishes open into a single chamber covered by a hard flap. Movement of the flap improves the flow of water over the gills, where gas exchange takes place. Early rayfin fishes also evolved gas-filled sacs that supplemented the action of the gills in respiration. These features enabled early rayfin fishes to live where oxygen was periodically in short supply, as it often is in freshwater environments. The lung-like sacs evolved into swim bladders, which function as organs of buoyancy in most rayfin fishes today. By adjusting the amount of gas in its swim bladder, a fish can control the depth at which it is suspended in the water without expending energy. Rayfin fishes radiated during the tertiary into about 24,000 species, encompassing a remarkable variety of sizes, shapes, and lifestyles. The smallest are less than 1 centimeter long as adults, the largest weigh up to 900 kilograms. Rayfin fishes exploit nearly all types of aquatic food sources. In the oceans they filter plankton from the water, grasp algae from rocks, eat corals and other colonial invertebrates, dig invertebrates from soft sediments, and prey upon virtually all other fishes. In freshwater they eat plankton, devour insects of all aquatic orders, eat fruits that fall into the water in flooded forests, and prey on other aquatic vertebrates and occasionally terrestrial vertebrates. Some fishes live buried in soft sediments, capturing passing prey or emerging at night to feed. Many fishes are solitary, but in open water others form large aggregations called schools. Many fishes perform complicated behaviors by means of which they maintain schools, build nests, court and choose mates, and care for their young. Although rayfin fishes can readily control their positions in open water, their eggs tend to sink. A few species produce small eggs that are buoyant enough to complete their development in open water. However, most marine fishes move to food-rich shallow waters to lay their eggs, which is why coastal waters and estuaries are so important in the life cycles of many species. Some, such as salmon, actually abandon saltwater when they breed, ascending rivers to spawn in freshwater streams and lakes. Colonizing the land, obtaining oxygen from the air. The evolution of lung-like sacs in fishes appears to have been in response to the inadequacy of gills for respiration in oxygen-poor waters, but it also set the stage for the invasion of the land. Some early rayfin fishes probably used their lungs to supplement their gills when oxygen levels in the water were low, as lungfishes do today. This ability would also have allowed them to leave the water temporarily and breathe air when pursued by predators unable to do so. But with their unjointed fins, these fishes could only flop around on land, as most fish out of water do today. Changes in the structure of the fins allowed these fishes to move on land. The lobefin fishes, class Actinistia, were the first lineage to evolve jointed fins. Lobefins flourished from the Devonian period until about 65 million years ago, when they were thought to have become extinct. However, in 1938, a living lobefin was caught by commercial fishermen off South Africa. Since that time, several dozen specimens of this extraordinary fish, Lagenaria chilonae, have been collected. Lagenaria, a predator on other fish, reaches a length of about 1.8 meters and weighs up to 82 kilograms. Its skeleton is mostly composed of cartilage, not bone. A second species, L. melanosis, was discovered in 1998 off the Indonesian island of Sulawesi. Lungfishes class Dipnoi were important predators in shallow water habitats in the Devonian, but most lineages died out. The three surviving species live in stagnant swamps and muddy waters in the southern hemisphere, one each in South America, Africa, and Australia. Lungfishes have both gills and lungs. When ponds dry up, they can burrow deep into the mud and survive for many months in an inactive state. It is believed that descendants of some lungfishes began to use terrestrial food sources, became more fully adapted to life on land, and eventually evolved to become the tetrapods, the four-legged amphibians, reptiles, birds, and mammals. Amphibians invaded the land. During the Devonian period, amphibians class Amphibia arose from an ancestor they shared with lungfishes. In this lineage, stubby, jointed fins evolved into walking legs. The basic design of these legs has remained largely unchanged throughout the evolution of terrestrial vertebrates. The Devonian predecessors of amphibians were probably able to crawl from one pond or stream to another by slowly pulling themselves along on their fin-like legs, as do some modern species of catfishes. They gradually evolved the ability to live in swamps and, eventually, on dry land. Modern amphibians have small lungs, and most species exchange gases through their skins as well. Most terrestrial species are confined to moist environments because they lose water rapidly through their skins when exposed to dry air, and because they require water for reproduction. About 4,500 species of amphibians live on Earth today, many fewer than the number known only from fossils. Living amphibians belong to three orders: the worm-like, limbless, tropical, burrowing caecilians order Gymnophiona, the frogs and toads order Neura, which means tailless, and the salamanders order Urodila, which means tailed. Most species of frogs and toads live in tropical and warm temperate regions, although a few are found at very high latitudes and altitudes. Some toads have tough skins that enable them to live for long periods of time in dry places. Salamanders are most diverse in temperate regions, but many species are found in cool, moist environments in Central American mountains. Many salamanders that live in rotting logs or moist soil lack lungs. They exchange gases entirely through the skin and mouth lining. Amphibians are the focus of much attention today because populations of many species are declining rapidly. Most species of amphibians live in water at some time in their lives. In the typical amphibian life cycle, part or all of the adult stage is spent on land, but adults return to fresh water to lay their eggs. Amphibian eggs can survive only in moist environments because they are enclosed within delicate envelopes that cannot prevent water loss in dry conditions. The fertilized eggs of most species give rise to larvae that live in water until they undergo metamorphosis to become terrestrial adults. Some amphibians, however, are entirely aquatic, never leaving the water at any stage of their lives. Others are entirely terrestrial, laying their eggs in moist places on land and skipping the aquatic larval stage. Amniotes colonize dry environments. Two morphological changes contributed to the ability of one lineage of tetrapods to control water loss and, therefore, to exploit a wide range of terrestrial habitats. Evolution of an egg with a shell that is relatively impermeable to water. A combination of traits that included a tough skin impermeable to water and kidneys that could excrete concentrated urine. The vertebrates that evolved both of these traits are called amniotes. They were the first vertebrates to become widely distributed over the terrestrial surface of Earth. The amniote egg has a leathery or brittle, calcium impregnated shell that retards evaporation of the fluids inside but permits O2 and CO2 to pass through. Such an egg does not require a moist environment and can be laid anywhere. Within the shell and surrounding the embryo are membranes that protect the embryo from desiccation and assist its respiration and excretion of waste nitrogen. The egg also stores large quantities of food as yolk, permitting the embryo to attain a relatively advanced state of development before it hatches and must feed itself. Figure 33. An early amniote lineage, the reptiles, arose from a tetrapod ancestor in the Carboniferous period. Figure 33. The class Reptilia, as we use the term here, is a paraphyletic group because some reptiles, crocodilians, are in fact more closely related to the birds than they are to lizards, snakes, and turtles. However, because all members of Reptilia are structurally similar, it serves as a convenient group for discussing the characteristics of amniotes. Therefore, we use the traditional classification of Reptilia as a basis for our discussion while recognizing that, technically, the birds should be included within it. About 6,000 species of reptiles live today. Most reptiles do not care for their eggs after laying them. In some species, the eggs do not develop shells, but are retained inside the female's body until they hatch. Some of these species evolve a structure called the placenta that nourishes the developing embryos. The skin of the reptile is covered with horny scales that greatly reduce loss of water from the body surface. These scales, however, make the skin unavailable as an organ of gas exchange. In reptiles, gases are exchanged almost entirely by the lungs, which are proportionally much larger in surface area than those of amphibians. A reptile forces air into and out of its lungs by bellows-like movements of its ribs. The reptilian heart is divided into 3 1/2 or 4 chambers that partially separate oxygenated from unoxygenated blood. With this type of heart, reptiles can generate higher blood pressures than amphibians, which have 3-chambered hearts and can sustain higher levels of muscular activity. Reptilian lineages diverged. The lineages leading to modern reptiles began to diverge about 250 millennia. One lineage that has changed very little over the intervening millennia is the turtles subclass Testudines. Turtles have a combination of ancestral traits and highly specialized characteristics that they do not share with any other vertebrate group. For this reason, their phylogenetic relationships are uncertain. The dorsal and ventral bony plates of modern turtles and tortoises form a shell into which the head and limbs can be withdrawn. Most turtles live in lakes and ponds, but tortoises are terrestrial. Some live in deserts. Sea turtles spend their entire lives at sea except when they come ashore to lay eggs. All 7 species of sea turtles are endangered. A few species of turtles and tortoises are carnivores, but most species are omnivores that eat a variety of aquatic and terrestrial plants and animals. The subclass Stomata includes lizards and snakes as well as the amphisbaenians, a group of legless, worm-like, burrowing animals with greatly reduced eyes. The Tuatara subclass Sphenodontia are a sister group to the lizards and snakes. Sphenodontids were diverse during the Mesozoic era, but today they are represented only by 2 species restricted to a few islands off the coast of New Zealand. Tuataras superficially resemble lizards, but differ from them in tooth attachment and several internal anatomical features. Most lizards are insectivores, but some are herbivores. A few prey on other vertebrates. The largest lizards, growing as long as 3 meters, are certain monitor lizards such as the Komodo dragon that live in the East Indies. Most lizards walk on 4 limbs, but some are limbless, as are all snakes, which are descendants of burrowing lizards. All snakes are carnivores. Many can swallow objects much larger than themselves. This is the mode of feeding of the largest snakes, the pythons, which can grow to more than 10 meters long. Several snake lineages evolved a combination of venom glands and the ability to inject venom rapidly into their prey. A separate diverging lineage led to the crocodilians, subclass Crocodilia, and to the dinosaurs. The crocodilians, crocodiles, caimans, gharials, and alligators are confined to tropical and warm temperate environments. Crocodilians spend much of their time in water, but they build nests on land or on floating piles of vegetation. The eggs are warmed by heat generated by decaying organic matter that the parents place in the nest. Typically, the female guards the eggs until they hatch. All crocodilians are carnivorous. They eat vertebrates of all classes, including large mammals. The dinosaurs rose to prominence about 250 millennia and dominated terrestrial environments for about 150 million years. During the time, virtually all terrestrial animals more than a meter in length were dinosaurs. Some of the largest dinosaurs weighed up to 100 tons. Many were agile and could run rapidly. The ability to breathe and run simultaneously, which we take for granted, was a major innovation in the evolution of terrestrial vertebrates. Not until the evolution of the lineages leading to the mammals, dinosaurs, and birds did the legs assume vertical positions directly under the body, which reduced the lateral forces on the body during locomotion. Special muscles that enabled the lungs to be filled and emptied while the limbs moved also evolved. We can infer the existence of such muscles in dinosaurs from the structure of the vertebral column in fossils and the capacity of many dinosaurs for bounding, bipedal two-legged locomotion. Several fossil dinosaurs discovered recently in early Cretaceous deposits in Liaoning province, in northeastern China, clearly show that in some small predatory dinosaurs, the scales had been highly modified to form feathers. One of these dinosaurs, Microraptor Way, had feathers on all 4 limbs, and those feathers were structurally similar to those of modern birds. Birds, more feathers and better flight. During the Mesozoic era, about 175 millennia, a dinosaur lineage gave rise to the birds subclass Aves. The oldest known avian fossil, Archaeopteryx, which lived about 150 millennia, had teeth, unlike modern birds, but was covered with feathers that are virtually identical to those of modern birds. It also had well-developed wings, a long tail figure 34.20b, and a furcula, or wishbone, to which some of the flight muscles were probably attached. Archaeopteryx had clawed fingers on its forelimbs, but it also had typical perching bird claws, suggesting that it lived in trees and shrubs and used the fingers to assist it in clambering over branches. Because the avian lineage separated from other reptiles long before Archaeopteryx lived, existing data are insufficient to identify the ancestors of birds with certainty. Most paleontologists believe that birds evolved from feathered terrestrial bipedal dinosaurs that used their forelimbs for capturing prey. Many remains of other early birds have been discovered in 120 to 125 million year old fossil beds in northeastern China. One of these birds, Confucius ornithontus, is known from hundreds of complete specimens. The males had greatly elongated tail feathers, which they probably used in communal courtship displays. Large numbers of individuals have been found together, as would be expected if many males assembled on communal display grounds, as some birds do today. Birds range in size from the 2-gram hummingbird of the West Indies to the 150-kilogram ostrich. Some flightless birds of Madagascar and New Zealand known from fossils were even larger. These birds were exterminated by humans soon after they colonized those islands. There are about 9,600 species of living birds, more than in any other major vertebrate group except ray finfishes. As a group, birds eat almost all types of animal and plant material. A few aquatic species have bills modified for filtering small food particles from water. Insects are the most important dietary items for terrestrial species. Birds are major predators of flying insects during the day, and some species exploit the food source at night. In addition, birds eat fruits and seeds, nectar and pollen, leaves and buds, carrion, and other vertebrates. By eating the fruits and seeds of plants, birds serve as major agents of seed dispersal. The feathers developed by some dinosaurs may originally have had thermoregulatory or display functions. Birds also use them for flying. Large quills that arise from the skin of the forelimbs create the flying surfaces of wings. Other strong feathers sprout like a fan from the shortened tail and serve as stabilizers during flight. The feathers that cover the body, along with an underlying layer of down feathers, provide insulation. The bones of birds are modified for flight. They are hollow and have internal struts for strength. The sternum breastbone forms a large, vertical keel to which the flight muscles are attached. These muscles pull the wings downward during the main propulsive movement in flight. Flight is metabolically expensive. A flying bird consumes energy at a rate about 15 to 20 times faster than a running lizard of the same weight. Because birds have such high metabolic rates, they generate large amounts of heat. They control the rate of heat loss using their feathers, which may be held close to the body or elevated to alter the amount of insulation they provide. The brain of a bird is larger in proportion to its body than a lizard or crocodile brain, primarily because the cerebellum, the center of sight and muscular coordination, is enlarged. Most birds lay their eggs in a nest, where they are warmed by heat from an adult that sits on them. Because birds have such high body temperatures, the eggs of most species hatch within a few weeks. The offspring of many species are ultrashell, hatch at a relatively helpless stage, and are fed for some time by their parents. The young of other bird species, such as chickens, sandpipers, and ducks, are precocial, can feed themselves shortly after hatching. Adults of nearly all species attend to offspring for some time, warning them of and protecting them from predators, protecting them from bad weather, leading them to good foraging places, and feeding them. The origin and diversity of mammals. Mammals class Mammalia appeared in the early part of the Mesozoic era, about 225 million years ago, branching from a lineage of mammal-like reptiles. Small mammals coexisted with reptiles and dinosaurs for at least 150 million years. After the large reptiles and dinosaurs disappeared during the mass extinction at the close of the Mesozoic era, mammals increased dramatically in numbers, diversity, and size. Today, mammals range in size from tiny shrews and bats weighing only about 2 grams to the endangered blue whale, which measures up to 33 meters long and weighs up to 160,000 kilograms, the largest animal ever to live on Earth. Skeletal simplification accompanied the evolution of early mammals from their larger reptilian ancestors. During mammalian evolution, some bones from the lower jaw were incorporated into the middle ear, leaving a single bone in the lower jaw. The number of bones in the skull also decreased. The bulk of both the limbs and the bony girdles from which they are suspended was reduced. Mammals have far fewer, but more highly differentiated teeth than reptiles do. Differences in the number, type, and arrangement of teeth in mammals reflect their varied diets. Skeletal features are readily preserved as fossils, but the soft parts of animals are seldom fossilized. Therefore, we do not know when mammalian features such as mammary glands, sweat glands, hair, and a four-chambered heart evolved. Mammals are unique among animals in supplying their young with a nutritive fluid, milk secreted by mammary glands. Mammalian eggs are fertilized. Mammalian eggs are fertilized within the female's body, and the embryos undergo a period of development, all gestation, within a specialized organ, the uterus, prior to being born. In many species, the embryos are connected to the uterus and nourished by placenta. In addition, mammals have a protective and insulating covering of hair, which is luxuriant in some species but has been almost entirely lost in whales, dolphins, and humans. In whales and dolphins, thick layers of insulating fat blubber replace hair as a heat retention mechanism. Clothing assumes the same role for humans. The approximately 4,000 species of living mammals are divided into two major subclasses: Prototheria and Theria. The subclass Prototheria contains a single order, the Monotremata, with a total of 3 species, which are found only in Australia and New Guinea. These mammals, the duck-billed platypus and the spiny anteaters, or echidnas, differ from other mammals in lacking a placenta, laying eggs, and having legs that poke out to the side. Monotremes supply milk for their young, but they have no nipples on their mammary glands. Rather, the milk simply oozes out and is lapped off the fur by the offspring. Members of the other subclass, Theria, are further divided. Monotremes supply milk for their young, but they have no nipples on their mammary glands. Rather, the milk simply oozes out and is lapped off the fur by the offspring. Members of the other subclass, Theria, are further divided into two groups. In most species of the first group, the marsupialia, females have a ventral pouch in which they carry and feed their offspring. Gestation in marsupials is short. The young are born tiny but with well-developed forelimbs, with which they climb to the pouch. They attach to a nipple, but cannot suck. The mother ejects milk into the tiny offspring until they grow large enough to suckle. The subclass Prototheria contains a single order, the Monotremata, with a total of 3 species, which are found only in Australia and New Guinea. These mammals, the duck-billed platypus and the spiny anteaters, or echidnas, differ from other mammals in lacking a placenta, laying eggs, and having legs that poke out to the side. Monotremes supply milk for their young, but they have no nipples on their mammary glands. Rather, the milk simply oozes out and is lapped off the fur by the offspring. Members of the other subclass, Theria, are further divided into two groups. In most species of the first group, the marsupialia, females have a ventral pouch in which they carry and feed their offspring. Gestation in marsupials is short. The young are born tiny but with well-developed forelimbs, with which they climb to the pouch. They attach to a nipple, but cannot suck. The mother ejects milk into the tiny offspring until they grow large enough to suckle. Once her offspring have left the uterus, a female marsupial may become sexually receptive again. She can then carry fertilized eggs capable of initiating development and replacing the offspring in her pouch should something happen to them. There are about 240 living species of marsupials. At one time, marsupials were found on all continents, but today the majority of species are restricted to the Australian region, with a modest representation in South America. One species, the Virginia possum, is widely distributed in the United States. Marsupials radiated to become terrestrial herbivores, insectivores, and carnivores, but no marsupial species live in the oceans or can fly, although some are gliders. The largest living marsupial is the red kangaroo of Australia, which weighs up to 90 kilograms. Much larger marsupials existed in Australia until they were exterminated by humans soon after they reached the continent about 50,000 years ago. Most living mammals belong to the second therian group, the eutherians. Eutherians are sometimes called placental mammals, but this name is not accurate because some marsupials also have placentas. Eutherians are more developed at birth than are marsupials, and no external pouch houses them after birth. The nearly 4,000 species of eutherians are placed into 16 major groups, the largest of which is the rodents order Rodentia with about 1,700 species. The next largest group, the bats order Chiroptera, has about 1,000 species, followed by the moles and shrews order Insectivora with slightly more than 400 species. Eutherians are extremely varied in their form and ecology. Several lineages of terrestrial eutherians subsequently colonized marine environments to become whales, dolphins, seals, and sea lions. Eutherian mammals are, or were, until they were greatly reduced in numbers by humans, the most important grazers and browsers in most terrestrial ecosystems. Grazing and browsing have been an evolutionary force intense enough to select for the spines, tough leaves, and difficult to eat growth forms found in many plants, a striking example of coevolution. Primates and the origin of humans. A eutherian lineage that has had dramatic effects on ecosystems worldwide is the primate lineage, which has undergone extensive recent evolutionary radiation. Primates probably descended from small arboreal tree-living insectivorous mammals early in the Cretaceous period. Anally complete fossil of an early primate species, Carpelus, from Wyoming, dated at 56 Maya, had grasping feet with an opposable big toe that had a nail rather than a claw. Such grasping limbs are one of the major adaptations to arboreal life that distinguish primates from other mammals. However, Carpelus did not have eyes positioned on the front of the face to provide good depth perception, as all modern primates do. Early in its evolutionary history, the primate lineage split into two main branches, the prosimians and the anthropoids, figure 35. Prosimians, lemurs, pottos, and lorises once lived on all continents, but today they are restricted to Africa, Madagascar, and tropical Asia. All of the mainland prosimian species are arboreal and nocturnal. However, on the island of Madagascar, the site of remarkable prosimian radiation, there are also diurnal and terrestrial species. The anthropoids, tarsiers, monkeys, apes, and humans evolved from an early primate lineage about 55 million years ago in Africa or Asia. New World monkeys diverged from Old World monkeys early enough that they could have reached South America from Africa when those two continents were still close to each other. All New World monkeys are arboreal. Many of them have long, prehensile grasping tails with which they can hold onto branches. Many Old World primates are arboreal as well, but a number of species are terrestrial. Some of these species, such as baboons and macaques, live and travel in large groups. No Old World primates have prehensile tails. About 22 million years ago, the lineage that led to modern apes separated from the other Old World primates. Between 22 and 5.5 Maya, as many as 100 species of apes range over Europe, Asia, and Africa. About 9 Maya, members of one European ape lineage, Dryopithecus, migrated to Africa and became the ancestors of the modern African apes, gorillas and chimpanzees, and of humans. The Asian apes, gibbons and orangutans, are a different ape lineage, descendants of Sauropithecus. Human ancestors evolved by pedal locomotion. The hominids, the lineage that led to humans, separated from other ape lineages about 6 Maya in Africa. The earliest proto-hominids, known as Ardipithecines, had distinct morphological adaptations for bipedalism, locomotion in which the body is held erect and moved exclusively by movements of the hind legs. Bipedal locomotion frees the forelimbs to manipulate objects and to carry them while walking. It also elevates the eyes, enabling the animal to see over tall vegetation to spot predators and prey. At walking rates, bipedal movement is also energetically much more economical than quadrupedal four-legged locomotion. All three advantages were probably important for the Ardipithecines and their descendants, the Australopithecines. The first Australopithecine skull was found in South Africa in 1924. Since then, Australopithecine fossils have been found in many sites in Africa. The most complete fossil skeleton of an Australopithecine, approximately 3.5 million years old, was discovered in Ethiopia in 1974. That individual, a young female known to the world as Lucy, was assigned to the species Australopithecus afarensis. Fossil remains of more than 100 afarensis have now been discovered. During the past 5 years, fossils of other Australopithecines that lived in Africa 4 to 5 million years ago have been unearthed. Experts disagree over how many species are represented by the Australopithecine fossils, but it is clear that several million years ago, at least 2 distinct types lived together over much of eastern Africa. The larger type, about 40 kilograms, is represented by at least 2 species, Paranthropus robustus and P. boisei, both of which died out suddenly about 1.5 million years ago. Humans arose from Australopithecine ancestors. Early members of the genus Homo lived contemporaneously with Australopithecines for perhaps half a million years, figure 36. The oldest fossils of the genus, an extinct species called H. habilis, were discovered in the Olduvai Gorge, Tanzania. These fossils are estimated to be 2 million years old. Other fossils of H. habilis have been found in Kenya and Ethiopia. Associated with the fossils are tools that these early hominids used to obtain food. Another extinct species of our genus, Homo erectus, evolved in Africa about 1.6 millia. Soon thereafter, it had spread as far as eastern Asia. As it expanded its range and increased in abundance, H. erectus may have exterminated H. habilis. Members of H. erectus were as large as modern people, but their bones were considerably heavier. Homo erectus used fire for cooking and for hunting large animals, and made characteristic stone tools that have been found in many parts of the Old World. Although H. erectus survived in Eurasia until about 250,000 years ago, it was replaced in tropical regions by our species, Homo sapiens, about 200,000 years ago. Human brains became larger. The earliest members of Homo sapiens had larger brains than members of the earlier species of Homo. Brain size in the lineage increased rapidly, reaching modern size by about 160,000 years ago. This striking change was probably favored by an increasingly complex social life. The ability of group members to communicate with one another would have been valuable in cooperative hunting and gathering and for improving one's status in the complex social interactions that must have characterized early human societies, just as they do ours today. But why did brains become larger only in the human lineage? A clue to the answer is provided by brain chemistry. The human brain is a fat-rich organ. About 60% of its structural material is made up of lipids, most of them long-chain polyunsaturated omega-3 and omega-6 fatty acids. Humans must consume omega fatty acids in their diet because the body cannot synthesize these molecules fast enough from the other fatty acids found in vegetables, nuts, and seeds to supply their brains. Animal brains and livers contain omega fatty acids, but fish and shellfish are by far the best sources. Therefore, because savannas and open woodlands provide few sources of omega fatty acids, the traditional view that early human evolution took place in those environments is being questioned. In contrast, the shores of Africa's many lakes would have been rich sources of fish and mollusks. Thus, access to fat-rich foods from aquatic environments may have been a key factor that supported the dramatic expansion of the human brain. The archaeological record of the past 100,000 years includes hundreds of piles of mollusk shells and fish bones, as well as hard points used for fishing. Chimpanzees remained in the forest and ate fruits and nuts. They may have lacked food sources to support much larger brains. Several Homo species existed during the mid-Pleistocene epoch, from about 1.5 million to about 300,000 years ago. All were skilled hunters of large mammals, but plants continued to be important components of their diets. During this period, another distinctly human trait emerged: rituals and a concept of life after death. Deceased individuals were buried with tools and clothing, presumably supplies for their existence in the next world. One species, Homo neanderthalensis, was widespread in Europe and Asia between about 75,000 and 30,000 years ago. Neanderthals were short, stocky, and powerfully built humans whose massive skulls housed brains somewhat larger than our own. They manufactured a variety of tools and hunted large mammals, which they probably ambushed and subdued in close combat. For a short time, their range overlapped that of the H. sapiens known as Cro-Magnons, but then the Neanderthals abruptly disappeared. Many scientists believe that they were exterminated by the Cro-Magnons, just as H. habilis may have been exterminated by H. erectus. Cro-Magnon people made and used a variety of sophisticated tools. They created remarkable paintings of large mammals, many of them showing scenes of hunting, that have been discovered in European caves. The animals depicted were characteristic of the cold steppes and grasslands that occupied much of Europe during periods of glacial expansion. Cro-Magnon people spread across Asia, reaching North America perhaps as early as 20,000 years ago, although the date of their arrival in the New World is still uncertain. Within a few thousand years, they had spread southward through North America to the southern tip of South America. Deuterostomes and protozoans shared evolutionary themes. The evolution of deuterostomes paralleled the evolution of protozoans in several important ways. Both lineages exploited the abundant food supplies buried in soft marine substrata, attached to rocks, or suspended in water. Many groups of both lineages developed elaborate structures for moving water and extracting prey from it. In some lineages of both groups, the body cavity became divided into compartments that allowed better control of shape and movement. Some members of both groups evolved mechanisms for controlling their buoyancy in water using gas-filled internal spaces. Planktonic larval stages evolved in marine members of many protostomate and deuterostomate phyla. Both protozoans and deuterostomes colonized the land, but the consequences were very different. The jointed external skeletons of arthropods, although they provide excellent support and protection in air, cannot support large animals, as the internal skeletons developed by deuterostomes can. Terrestrial deuterostomes recolonized aquatic environments a number of times. Suspension feeding evolved once again in several of these lineages. The largest living animals, baleen toothless whales, feed upon small prey only a few centimeters long, which they extract from the water with large straining structures in their mouths. Major habitat type of Indian subcontinent. This subcontinent has been affected worst by human influences since long line. Due to this, climax formations are very much altered and/or destroyed for agriculture and other similar purposes. The present vegetation has suffered immensely from effects of plants, animals, soil, climate, and man. Therefore, the vegetation of the country that we see around us are much interfered. The most important factors used in the classification of vegetation are rainfall, temperature, biotic influences, and life forms. In India, there are two most common types of plant formations: high forest and deep grassland. Forest vegetation. Indian forests have generally been classified on the basis of temperature into four major types: I tropical, E montane subtropical, E temperate, and IV alpine. Tropical forests. Common in the warmer plains, ranging from very dense, multi-storied of diverse trees, shrubs and lianas in areas of high rainfall to dry, scrub jungles of thorny bushes and isolated patches in dry areas. Thus, there are two types: moist tropical and dry tropical forests. Moist tropical forests. On the basis of degree of wetness, these are the following three principal types. High tropical wet evergreen forests, where annual rainfall is over 250 centimeters, as in West Coast, Assam, Bengal, and Andaman Islands. They are multi-storied, made up of small trees, shrubs, epiphytes, lianas, and dense brown vegetation. The dominant members are such trees as Dipterocarpus, Hopi, Artocarpus, Mangifera, Emblica, Michelia, Arbanea, Lagerstroemia, Ixora, and some climbers. Deep tropical moist semi-evergreen forests, better developed in northern and the southern region of country. Trees shed their leaves for brief period. In north, they develop in North Assam and Bengal and parts of Orissa receiving heavy rainfall. There are some evergreen plants also as Artocarpus, Michelia, and Eugenia. The deciduous trees are Terminalia, Terminals, and Chiria. Deep tropical moist deciduous forests, in which some trees shed leaves for brief periods, some are evergreen and semi-evergreen. They are common in moist areas of Kerala, Karnataka, and South MP and southern parts of North UP, MP, Bihar, Bengal, and Orissa in north. The well-known teak and sal forests belong to this category. In South India, moist deciduous forests are dominated by species of Terminalia, Grullia, Garya, Salmolia, Tectona grandis, Adina cordicolia, Melia, Albizia, Dalbergia, etc. In the northern half, Sharia robusta is dominant in Gorakhpur and terai regions of UP, Assam, and North Bengal. Other associates of Sal are Terminalia tomentosa, Dillenia sp, Eugenia sp, etc. Dry tropical deciduous forests. Most of their trees remain leafless for several weeks in dry season. In north, such forests are found in the Punjab, UP, Bihar, and Orissa in the regions which are neither wet nor too dry. Trees are of moderate size, 2.5 meters tall, with sparse canopy. Thorny scrubs, grasses, and some bamboos are also present in some regions. In Punjab and West UP forests, Onophysis latifolia, Acacia catechu, Terminalia tomentosa, Boswellia serrata are dominant with subdominance of Dendrocalamus strictus, Emblica officinale, Woodfordia floribunda, etc. Forests of Sharia robusta are also found scattered in wet areas. In south, such forests are trees with scattered patches of densely grown grasses intermixed with shrubs. Terminalia. Onophysis latifolia, Tectona grandis, Diospyros melanoxylon, Boswellia sp. Fawn the top layer. Followed by smaller plants like Dendrocalamus, Bambusa, Lantana, Helicteris, etc. Common grasses are Panicum, Andropogon, and Heteropogon. Montane subtropical forests. These are found on hills of South India, as Nilgiri, Mahabaleshwar, and Pachmarhi, between an altitude of 3,000 to 5,600 feet. These are cooler than the tropical and warmer than the temperate forests. In southern parts, the common trees are Eugenia, Actinodaphne, Campium, Mangifera, and Ficus, and climbers are Piper trichostachian, Nenum scandens, Smilax macrophylla, etc. The northern areas have rather tailed trees. In eastern Himalayas, due to higher humidities, bamboos, many epiphytes including orchids, and ferns become abundant. Most of the trees are evergreen. The floristic description of eastern and western Himalayas has already been given. Temperate forests. They occur above 5,300 feet altitude, chiefly on mountains of Himalayas and Nilgiri. In Himalayas, oaks and conifers are abundant. Their distribution has already been described. Oaks spawn relatively stable evergreen pure stands. The southern temperate vegetation is chiefly represented by the Cholas near Udugamand, Nilgiri hills in Tamil Nadu. The forests are very dense with extensive growth of grasses and evergreen tall trees, like Balanocarpus utilis, Hopiparviflora, Artocarpus hirsuta, Salmonia malvaricum, etc. Their branches are clothed with mosses, many woody climbers, ferns and other epiphytes. Alpine forests. Sometimes they are subdivided as subalpine forests, alpine scrubs, moist alpine scrubs and dry alpine scrubs. They are extensive throughout the Himalayas above 11,000 feet. The tree height becomes lesser with increasing altitude, being replaced finally by sparse